So, hello students and my dear future doctors, welcoming you again to this platform, to our channel, that is PW NEET English. And today is the day we'll be completing two chapters from class 12 biology. One is human reproduction and another is reproductive health. Okay, so how you how all are doing and your preparation? What about your preparation for upcoming NEET exam, or you are studying in class 11th or class 12th? Okay, so if you are studying in class 12 and you are an aspirant for NEET, then these two chapters are very much important for all of us. Obviously, these two are high weighted chapters from human reproduction and reproductive health. If you include both the chapters, minimum 6 to 7 questions can be expected, right? So let's start. Hello Swathik. So Swathik, how are you? Rudayanjali, yes, good afternoon, good evening. So how are you all? Can you write down in the comment section from where you people are watching? Swatik, from where you are, and Rudayanjali, you are watching from which place? Okay, so gradually I'll be moving forward to the first chapter of today, that is human reproduction. Okay, so you are Swatika, not an issue. Yeah, Tejwashini, yeah, hello, very good evening. Okay, Rudanjali, so you are from AP, Andhra, that's great. So let's start with the chapter. So when you talk about human reproduction, so what kind of animal are we? So basically, we will be reproducing through sexual mode of reproduction. You haven't seen any human being who is performing asexual mode, right? We are continuous breeder, means any time of the year we can perform this phenomena of reproduction. And obviously the mode of fertilization is internal for us, because the fusion of male and female gamete will be happening inside the female reproductive tract, as well as the development of the baby will be taking place inside the mother's womb. Okay, so Swathika, you are from Hyderabad, Telangana. Okay, that's great. So if you are aware about this fact that we are sexually reproducing and viviparous animals, because we are mammals, and viviparity means giving birth of young ones directly. Those animals who are laying eggs, we call them oviparous, right? So can you please write down one of the example of mammalian animal but which will be exhibiting oviparity? Can you write down in the comment section? We are obviously viviparous in nature. Can you name a mammal which is oviparous in nature? Right here in the comment section. Okay, so if we are moving forward, our reproductive events, if we talk about our reproductive events, we have to talk about gametogenesis, insemination, fertilization, implantation, gestation, and the termination will be through the process of parturition, that is called as childbirth. So what do you mean by gametogenesis? The process of formation of gametes. The process of formation of gametes means sperms and ovum. So that's the process, we call it gametogenesis. Yeah, exactly, Tejoshini given the right answer. It's ornithorhynchus, it's an oviparous mammal. Rudanjali also given the right answer. Monotremes, that's great, Ishika. Okay, so why gametogenesis is important? Because if we check our somatic cells, they are diploid in nature. Each and every cells in our body, they are possessing 46 chromosome. But the new zygote to be formed, that should also possess 46, or diploid sets of chromosome. That's why while we are forming a male gamete or female gamete, directly the chromosome number is halved. So that is the importance of gamete formation, as because these gametes are haploid in nature. Each gamete is having 23 numbers of chromosomes. And the next process is insemination. What do you mean by insemination? The transfer of male gametes into the reproductive tract of female. So that's the process of insemination. Transfer of sperms into the reproductive tract of female. That mechanism is called as insemination. And third, that is fertilization. Obviously, it's very easy. It's the fusion of male and female gamete, fusion of sperm and ovum is called fertilization. And upon fertilization, what we can observe? We can observe formation of a diploid zygote, right? Upon fertilization, and gradually the zygote will flourish. The zygote will start its cell division. We call it cleavage. Yes, very good evening, Surya. So this zygote will be going through various phases called morula and blastocyst. Now this blastocyst phase is gonna get a temporary attachment for 9 months with the endometrium or inner lining of uterus, that is called implantation. So what is implantation? Is the attachment of blastocyst. Attachment of blastocyst. This is a very important, a very crucial stage of embryo, right? So the attachment of blastocyst in the uterine endometrium, means the inner lining of uterus, is called implantation. Now, can you please write down in the comment section who knows that which stage of embryo is called as blastocyst? Can you answer the question? Which stage of embryo is called as blastocyst? Or sometime we use the terminology as blastula. Okay. So when you are talking about implantation, we need to think about blastocyst, because this is the specialized stage, a football-like stage of embryo which is getting embedded into the inner layer of uterus, that is called uterine endometrium. And the next phase is called gestation, because basically this gestation is the development of the embryo will be happening here. The embryogenesis and the morphogenesis is taking place. This is also called as pregnancy period. So the pregnancy period is called gestation, and for us the gestation period is somehow nearby 7 months to 9 months is our gestation period. And at the end, at the terminal phase of this gestation period, there will be childbirth or the delivery of that baby, and that is called parturition. So parturition is nothing but childbirth. Okay, so no, my dear Ishika, it's not blastocyst is not 8-cell stage of embryo. 8-cell and 16-cell stage of embryo is called morula. Okay, blastocyst, when the 16-cell stage will be dividing, now starting from 32 to near about 128 cell stage, that is called as blastocyst, which is kind of a hollow football kind of structure. So blastocyst is having more than 16 cells. It's 8-cell stage and 16-cell stage, that is called as morula, remember. Okay. So If we talk about our reproductive events, there are remarkable differences between the reproductive events in the male and in the female. For example, sperm formation, that is called spermatogenesis, continues even in old men. Okay, there is beginning of spermatogenesis, but there is nothing called spermiopause, right? Throughout the life, man can produce sperm inside the seminiferous tubule of testes. But in case of female, there is formation of ovum, that is called as oogenesis. Oogenesis, that will be ceasing, that is ceases in woman around the age of 50 year, and you know that is called as menopause. So for female, there is a menopause near about 45 to 50 years of age. There will be some drastic change in the hormone, and the whole phenomena of menstruation and ovulation will be stopped. So that is called menopause. But for male, there is nothing is called spermiopause, right? So that is the basic difference between the phenomena in male and female. There is one more thing, one more important thing. Spermatogenesis in case of male will be beginning at the age of puberty, at the age of 11 years, 12 years or 13 years, right? The beginning of spermatogenesis. But in case of female, oogenesis, it will be beginning during the time of the embryonic developmental phase, when that girl child is in fetus form, staying inside the mother's womb or mother's uterus. Then only her little ovaries will be initiating the process of oogenesis. That is another remarkable difference between the reproductive event of male and female. Okay. Now, we are entering into the reproductive system. We will be talking about male reproductive system and female reproductive system, but the common thing is that for both the cases, there will be a set of primary reproductive organs. Like in case of male, it's one pair of testes, and in case of female, that is one pair of ovaries, right? Now, what is the function of primary reproductive structure or primary reproductive organ? You need to remember this. Either we talk about testes or you talk about ovaries. Both of them are called as primary reproductive organs or gonads, and their major function is to do gametogenesis. Gametogenesis for testis, sperm formation; for ovaries, ovum formation; and second, production of sex hormones. Production of sex hormones. In case of male, it's androgen or testosterone, and in case of male, ovaries will be producing estrogen, progesterone, relaxin, and few other hormones. So these two are the remarkable function carried out by primary reproductive organs. You can call them as gonads. Remember, primary reproductive organs are also called as gonads. So testis is the gonad of male and ovary gonad of female. Now, what is the function of secondary reproductive organs? See, secondary reproductive organs, like in case of male, the accessory ducts and glands are called as secondary reproductive organs. In case of females, is similar. The accessory ducts and glands are called as secondary reproductive organs. So, what is the function that is played by secondary reproductive organ? The nourishing. They will be providing nourishment to the gametes. Secondary reproductive parts will not be producing gametes. Secondary reproductive part will not be producing any sort of sex hormones. Rather, they will provide the place for fusion of sperm and ovum. So that is the importance of secondary reproductive organ. They provide nourishment to the gametes. They provide nourishment and help in their survivability, viability, and provide the space or place for fertilization. That is the importance of secondary reproductive organs. Okay. Now we'll jump inside the anatomy of male reproductive system. Yes. So the male reproductive system is located in dash region. So which part of our body you will be finding male reproductive system? So the location will be pelvic region, or you can say lower abdominal region. So pelvic region is also called as lower abdominal region. So male reproductive system is present into lower abdomen or pelvic region of our body. And as we were talking about, male reproductive system is divided again into primary reproductive structures and secondary reproductive structure. When you are talking about primary reproductive organ, we have one pair of testes or two testes, and function is to do sperm formation or spermatogenesis and production of male sex hormone, that is called as androgens. You know about testosterone, right? So testosterone is the prime androgen produced by our testes. Now, let's talk about secondary reproductive structures into male bodies. So there are some accessory ducts, some accessory glands, and obviously external genitalia. So what are the external genitalia associated with the secondary reproductive structure? There are two external genitalia present into male reproductive system. One is penis and another the sac, the specific sac like structure, the skinny sac in which both the testes are present, that is scrotum. So penis and scrotum, these two are regarded as external genitalia. What are the accessory glands situated inside our body which are part of reproductive system? They are seminal vesicles. Seminal vesicles. There are two seminal vesicles. Okay, prostate gland, one prostate gland, and two Cowper's gland or bulbourethral gland. Two Cowper's glands, or these Cowper's glands are also called as bulbourethral gland. Yes. Now, what are the accessory ducts? What are the accessory ducts present into a male reproductive system? Okay, so they are like this: tubuli recti, tubuli recti, then reti testis, then vasa efferentia, then epididymis, and then vas deferens. Vas deferens. Then we have ejaculatory duct. And lastly, the last part of accessory duct, that is called urethra. Also for male, it's called as urinogenital duct. So these are the parts of accessory ducts. Who are they? They are tubuli recti, reti testis, vasa efferentia, epididymis, vas deferens, ejaculatory duct, and urethra. And you have to follow this chronological order. Yes. Now, so these two are the diagrams of male reproductive system. One is the frontal view and one is the sagittal view or lateral view. So we have to point out the different important structures here. So let's start with the primary reproductive organ, that is testes. Okay. So testis, and testis is protected, well protected inside the skinny sac called scrotum. Testis is well protected inside the skinny sac called scrotum, and outside the testis will be getting highly coiled tubes. These are called epididymis. Outside the testis, you will be getting highly coiled tubes, and these structures are called as epididymis. Okay. Now, the duct which is moving upward and going towards the abdominal cavity, that is the vas deferens. This vas deferens will be getting inside the abdominal cavity, and it will take a U-turn or looping nearby the urinary bladder. This is the urinary bladder. So, nearby this urinary bladder, the junction of ureters and this urinary bladder, this vas deferens will take a U-turn, and again it will be descending downwards. Okay. Now, while it's descending downwards, this vas deferens will merge with the ducts coming from seminal vesicles. So, these two glands are called seminal vesicles. Here also you can observe the seminal vesicle. The vas deferens will merge with the seminal duct and will form a common duct called ejaculatory duct. So, what is ejaculatory duct? Ejaculatory duct is a merged version of vas deferens and seminal duct. Okay. Now, this ejaculatory duct will come forward and will get merged with the duct coming from prostate and with the duct coming from the urinary bladder, that is called urethra, and will be forming the urinogenital duct. Okay. Now, while this urinogenital duct is entering into the penis, it will get secretion of two more glands, and these two glands are called as Cowper's gland or bulbourethral gland. These two glands are called as Cowper's gland or bulbourethral glands. Cowper's gland or bulbourethral glands. Okay. Now, upon getting the secretion of Cowper's gland, urethra or urinogenital duct will enter into the penis, and the opening through penis of this urethra is called urethral meatus or urethral opening. Urethral meatus or urethral opening. This urinogenital duct is again called as urethra. Okay. Now, this is the penis, the copulatory organ of males. Copulatory organ means with the help of penis, a male will be inseminating the semen or the sperm inside the female reproductive system. So, that is all about the anatomical parts or anatomical structure of male reproductive system. Now, we will get inside the testes, the primary reproductive organ. So, when you are talking about testes, both the testes, they are situated within a pouch of scrotum. So, you know that the scrotum present outside the body, outside the abdominal cavity. So, these testes can be said as they are extra abdominal organs. Testes, they are extra abdominal organ, as because they are not present inside the abdominal cavity, they are present outside the abdominal cavity. Both the testes, they are present outside the abdominal cavity, they are not present inside the abdominal cavity. But during the time of embryonic developmental phase, in the beginning, one pair of testes will be produced, will be formed inside the abdominal cavity only. But with the help of spermatic cord, with the help of a cord called spermatic cord, both the testes will be descending down and will get stable inside the scrotal sac, inside that skinny pouch. And what is the reason behind that? Why our tests are not present inside the abdomen, present outside the abdomen? The reason is that the scrotum maintains the low temperature, that is 2 to 2.5 degrees Celsius less than our body temperature, and that is required for spermatogenesis. So that is the scientific reason why we don't have testes present inside our abdominal cavity. But for few person, for few male, this testis will be failing to descend down into the scrotal sac, and when they are matured, still they have testis present inside their abdominal cavity, and that particular terrific condition is called as cryptorchidism, when testis is present inside the abdominal cavity into a mature or adult man. And if you are suffering from cryptorchidism, then you will not be able to produce sperm, as because this temperature is very much crucial for sperm formation, that is near about 34 to 35 degrees Celsius. Okay. And as because testes, they are quite very sensitive organs, you need to cover them with proper covering, and the covering of testes, the main covering of testes is called as tunica albuginea, though testes is having 3 coverings. But in our NCERT, if we follow our NCERT, there is only one major covering is given. The main covering of testes is called as tunica albuginea, and the meaning of tunica is covering. Tunic means covering, remember this. Okay. Now if we talk about some dimension of testis, the length of testis is 4.425 centimeter, and width of testis is nearby 2 to 3 centimeter. Okay. And if you get inside one testis, that single testis is subdivided into 250 testicular lobules, one testis is having 250 testicular lobule. So if you are asked that how many testicular lobule present in human being, so inside human being there are 2 tests and 500 testicular lobules you have to answer, and inside each testicular lobule, means inside each chamber of testis, there will be 1 to 3 seminiferous tubules, and these are the special structures, seminiferous tubule, where sperm formation is going on. Right. Okay. Now if you get inside the seminiferous tubule, these are the tiny tubes where sperm is getting produced. If you get inside the seminiferous tubule, you will be finding lot of male germ cell. Now what do you mean by male germ cell? These are the cells which will be proliferating and dividing further to produce sperm. And also inside the seminiferous tubule, you are going to get some slender pillar like columnar cells, and we call them Sertoli cell. And as because these Sertoli cells are responsible for providing nourishment to this germ cell, these Sertoli cells also can be called as nurse cell. The Sertoli cells also can be called as nurse cell, and some people may call it sustentacular cell or sustenocyte. So if you are asked what kind of cells you are going to find inside each seminiferous tubule, so the answer will be, you are going to get male germ cells. These are the cells which are responsible for sperm formation, and we need to provide nourishment to these. So for that we have some columnar cells. Obviously the Sertoli cells are nothing but columnar cells. These are responsible for providing nutrition to this germ cell. So another name they got as nurse cell. Now let's move forward to check the anatomy, inside anatomy of testes. So if you take a section of testis, then the outer skinny covering is called, that is, scrotum. Skin of scrotum. Okay. Now, this is the first layer of covering, the first layer of protective tunic or protective covering, and that is simply called as, you don't have to study about that outermost covering of testis, right? That is called as tunica vaginalis. So the middle layer of this whitish covering, which is surrounding the main part of testis, that is actually our tunica albuginea. It is actually our tunica albuginea, and it's the tunica albuginea which is responsible for formation of this compartment or chambers inside our testes. And the innermost layer, in the innermost layer of testes, that is called tunica vasculosa, because this is highly vascular. So verbally I am telling their name. The outermost covering of testis, that is called tunica vaginalis. The middle layer of testis, that is called tunica albuginea, and this is the most important testicular covering. And the innermost layer of testis, that is called as tunica vasculosa. But you have to remember only about this tunica albuginea, because this is also responsible for what? This is also responsible for formation of the compartments inside each testis. You know that every testis is divided into 250 chambers called testicular lobules. So this is one testicular lobule. This is one testicular lobule. Like this, each testis is having 250, and inside the testicular lobule, this highly coiled tubular structure you are observing, these are called as seminiferous tubule. These are called as seminiferous tubule. And you know that inside these seminiferous tubules, spermatogenesis is going on. And how many seminiferous tubule is present inside each testicular chamber? Each testicular lobule may have 1 to 3 seminiferous tubule. Means possibility is there, you may get only one seminiferous tubule, you may get two seminiferous tubule, or even sometime you may get three seminiferous tubules into each testicular lobule. So overall we can say that every testis is possessing nearby 500 to 700 seminiferous tubules inside it, right? Okay. And the space outside the seminiferous tubule, that is called as interstitial space. Interstitial space. The space outside the seminiferous tubule, that is called interstitial space, and in this interstitial space you will be finding some very important cell for you. They are called as interstitial cell or Leydig cell, which are responsible for formation of the male sex hormone called testosterone. So the area outside the seminiferous tubule is called interstitial space, and inside this interstitial space we will be finding some important cells, and they are called as interstitial cells or Leydig cell, and these cells are responsible for formation of testosterone hormone, that is the prime male androgen, right? Okay. Now let's talk about other tubular parts. Now you can check here that all these seminiferous tubules are coming together and forming a network like structure, right? So this network like structure is called as retitestis. This network like structure is called as retitestis, and from retitestis 12 to 15 ducts are going outward, and they are called as vasa efferentia. and all these vasa efferentia together forming this highly coiled tubular structure that is called as epididymis. And if you check the epididymis, you will be getting three portion of epididymis. The upper portion of epididymis is called as head of epididymis or cap of epididymis or capute epididymis. The main part of epididymis is called body of epididymis or corpus of epididymis, and the last part of epididymis is called tail of epididymis or cauda epididymis. And from this tail part of epididymis, you will find the beginning of the sperm duct, which we call vas deferens. So here the question is asked that which part of epididymis, from which part of epididymis there is beginning or starting of vas deferens is there. So the answer will be its tail part of epididymis or from cauda part of epididymis, right. So this was all about the anatomy, the internal structure of a single testis. Now let's get into the theoretical part. So whatever I was telling, so the region outside the seminiferous tubules, that is called as interstitial space, and in NCERT it's perfectly written what kind of structures you are gonna get inside this interstitial space. You are gonna get some small blood vessel. Remember, they are not large blood vessel, they are small blood vessel. You may get statement based question, you may get any sort of question. Now if you check that it's written as inside interstitial space large blood vessels are present, then you have to mark it false. Remember this. Okay, inside interstitial space the most important, the star cells of interstitial space, they are the Leydig cell or interstitial cell, and you know that these Leydig cells are responsible for testosterone formation and some immunologically competent cells. These are nothing but WBC. Few WBC present inside this interstitial space, we call them immunologically competent cell. They are kind of macrophage, right. Okay, so it's written here Leydig cells or interstitial cells, they are same thing only. They synthesize and secrete testicular hormone. They are called as androgens. And remember, these are the androgen group of hormone, and the prime androgen is called as testosterone. Generally we say male sex hormone is testosterone, but testosterone belongs to the group of male sex hormone. They are called as androgen group. And if we talk about the classification of these androgens or testosterone, they are steroid hormones. They are steroid hormones, right. So testosterone is androgen and androgens are steroid kind of hormone. Remember, even all the sex hormones, for female also we will be talking about progesterone, estrogens, they are also steroid hormone. Okay, now this diagram is given in NCERT, the histological cross section of testis. So that is amplified and drawn by me here. So here you can check this circular part. Here I have shown one seminiferous tubule, and the space outside the seminiferous tubule, this space is called as our interstitial space. And in interstitial space, this particular cells, this group of cells, you can observe these are called as Leydig cells. These are called as Leydig cells or interstitial cell, and they are secreting testosterone hormone, right. Okay, now if you get inside the seminiferous tubule, this column shaped cells, these are called as Sertoli, and this cube or this oval shaped cells, these are called as germ cell. Sometime we call them spermatogonia when they are going to do spermatogenesis process. Okay. Now I'll talk about hormonal regulation of male reproductive system. Now, yes, RBC King who are coming after bio examination. Okay. Sir, what is the work of immunologically competent cell? Okay, I missed that question. Now, right now, I saw that. So, Ishika, immunologically competent cells, they are nothing but they are WBC, right? So, where you'll be finding them? Into the interstitial space. Into the interstitial space, these WBCs, these white cells, these are the immunologically competent cells. I have written as ICC, right? So, what is the function of WBC? Providing immunity, clearing dirt and debris from this interstitial space of testis. So, that is the simple function of this immunologically competent cell. Mostly these are macrophages, right? And you know that what are the functions of macrophage. Okay. Any more question? Any more question? Till now, whatever I taught, if you want to ask any sort of question, then you can ask freely. You can write your question in the comment section. So, hormonal regulation, why it's very important topic. See, while we are discussing about male reproductive system, reproduction phenomena in male, then it's regulated by our hormones, and these hormones again can stimulate or can regulate our nervous system, right? So, how it's happening? See, we know that who is master of master? Who is master? Now, few people will answer me that Thalapathy is the master, right? But pituitary is master of our body, and hypothalamus is controlling the master. That's why hypothalamus is called as master of master. Everything begins from here, hypothalamus. So, hypothalamus will be producing a special hormone called GnRH, that is called as gonadotropin releasing hormone. So, this gonadotropin releasing hormone will carry the message to the anterior pituitary, carry the signal coming from hypothalamus, and will tell anterior pituitary, please start secretion of gonadotropins. Now, which are gonadotropins hormones? There are two gonadotropins secreted into our body by our anterior pituitary, LH and FSH. What is full form of LH? LH is called as luteinizing hormone and FSH is called as follicle stimulating hormone. Now, in case of male, this LH will come to Leydig cell of your testis. This LH will come to Leydig cell of your testis and will tell Leydig cells you start secretion of androgen hormone or testosterone. So, ultimately, if you are asked that testosterone or Leydig cells are under influence of which gonadotropin hormone, then you have to answer it's LH. Okay. Now, what is the function of FSH? We have studied that inside the seminiferous tubules, we got some pillar-like cells, some columnar cells. These are called Sertoli cell. So, FSH gonna influence these Sertoli cells, and under the influence of FSH, Sertoli cell will release ABP, that is called as androgen binding protein. Now, what is the function of this androgen binding protein? See, from this Leydig cells, androgens are released. So, you need to give some invitation to this androgens or this testosterone that testosterone will be coming inside the seminiferous tubule to stimulate the process of sperm formation. So this is the invitation card called as androgen binding protein released by Sertoli cell, and by the influence of ABP, testosterone or androgens are gonna get inside the seminiferous tubules to produce, to stimulate the process of sperm formation, right? Okay. Now, SMF, another important hormone released by Sertoli cell, that is called sperm maturation factor. Without sperm maturation factor SMF, the sperms produced inside the seminiferous tubules will not be maturing, will not get structural maturity. So these two are very important hormones secreted by Sertoli cell, and Sertoli cell releases another hormone called as inhibin, and by the name you can guess it that it's inhibiting something. So if a male body is producing a high amount of testosterone and high amount of sperm, then you need to give a negative signal to your body, stop production. So in that case, this inhibin will give a negative feedback or will inhibit the secretion of FSH. So these are the hormones and their function, which are secreted by Sertoli cell. So remember in one sentence that LH hormone will be influencing Leydig cell, and that will be produce testosterone hormone. FSH, that will be influencing Sertoli cell. So that was all about the hormonal regulation of male reproductive system. Now from this topic, if you want to ask any question, you are free to ask into the chat box, into the comment section. From this hormonal regulation of male reproductive system part. Okay, now we'll discuss about male accessory glands. So three kinds of glands are present into male reproductive system, and you know their name. They are seminal vesicle, prostate gland, and bulbourethral gland, right? How many seminal vesicles are there? Two seminal vesicles, single one prostate gland, and two bulbourethral gland. And you know that the name of another name of bulbourethral gland is called as Cowper's gland. Okay, that's great, Ishika. Thank you very much. Okay, so another name of bulbourethral gland is called as Cowper's gland. Now, if you ask, sir, what is the function of seminal vesicles? See, seminal vesicles, these are the glands which are responsible for production of a liquid called as seminal fluid. Seminal fluid. So liquid produced by seminal vesicles are called seminal fluid, and the seminal fluid, they are rich in fructose. Seminal fluid, these are rich in fructose. Now, why I have uttered the name of fructose? Fructose is the main energy source for the spermatozoa or for sperms. Sperms will be eating this fructose, right? So whenever you are feeling weak, you are drinking glucose, right? But when the sperms are feeling weak, they need to respire, they need to gain energy, so they will be utilizing the fructose present into the seminal fluid, which is produced by seminal vesicle, right? Okay. And prostate will be secreting also some fluid called as prostatic fluid. Fluid produced by prostate is called prostatic fluid, and fluid produced by bulbourethral gland, they are nothing but mucus. Bulbourethral gland secretes alkaline mucus. Now if you combine all these fluids together, that is seminal fluid from seminal vesicle, prostatic fluid from prostate, and alkaline mucus mucus from bulbourethral gland, the fluid you are going to get, that is called as seminal plasma. So here please focus for once, this seminal plasma and seminal fluid, these two are two different entity, right? Seminal fluid is the secretion of seminal vesicle, but seminal plasma is the combination of secretion by seminal vesicle, prostate gland and bulbourethral gland. And overall this seminal plasma is alkaline in nature. Seminal plasma is alkaline in nature, and inside this seminal plasma, you need to keep your sperm. Without the seminal plasma, sperms will not be surviving, right? Okay, so the seminal plasma, it's written here, that's why they are rich in fructose, the food source for sperm. They are rich in calcium. The calcium salts, they are responsible for making the plasma alkaline, making the liquid alkaline, and also it's rich in with some enzymes. Now NCERT is saying that if you talk about the secretion of bulbourethral glands, just alkaline mucus, they help in lubrication of penis. The secretion of bulbourethral gland help into the process of lubrication of penis, that will be making the way smoother and neutral when the sperms are coming out of your penis during the time of insemination. So that is the importance of mucus secreted by bulbourethral gland. Okay, now we'll talk about external genitalia. Now already you had this, we were discussing external genitalia means one penis and one scrotum. Okay, now when we'll be talking about penis, it's made by a special erectile tissue. Whenever a male is getting sensation, right, during the time of coitus or sexual intercourse, so due to the presence of this special erectile muscle, there will be erection, that they are responsible for strengthening the penis, and this erectile muscle also helping the penis during the course of the process of insemination. So here the names are given of this erectile muscle: corpora cavernosa and corpus spongiosum. Now there are two corpora cavernosa and one corpus spongiosum muscles are present inside one penis. Okay, now if you check the enlarged end of penis, that is called as glans penis, and there is a loose fold which can cover the penis, which is retractile in nature, that is called foreskin. Now if you observe the structure of a penis, the retractile skin, that is called foreskin, or you can also call it pipreus. The bulged or the enlarged end is called as glans of penis or glans penis. Okay, now the here towards the dorsal surface of penis, this special tissue is present, that is called as corpora cavernosa. Surrounding the urethra, the spongy tissue is present, that is called as corpus spongiosum. And you can see here, through corpus spongiosum, urethra is coming out. Through corpus spongiosum, urethra is coming out, and through corpus spongiosum, you can observe the opening of urethra or the opening of penis called as urethral meatus. Okay, Prajwal, you are in second PU, got it. So we are done with the structure of penis. Now I got a question here. Let me check how many of you can answer into the comment section. So seminiferous tubules contain dash cells for providing nutrition to sperm. Please, if you know the answer, you can give your answer directly into the comment section. Which cells are providing nutrition to sperm cells inside male reproductive system? Nobody knows the answer or what? Okay, Ishika has given the answer and the answer is Sertoli cell. Exactly. It's Sertoli cell, the tall and slender cells which are responsible for providing nourishment to maturing sperm. Okay, another question here we got, assertion and reason. Assertion is saying the testes are located outside the abdominal cavity within the scrotum, and reason is saying the scrotal muscles help regulate to lower the temperature in the testes, essential for spermatogenesis. Now can you answer this question? Yes, Sharmila Devi also given the correct answer. Hi Sanskar, how you are doing? Can you answer this question? Okay, so Prajal has given the answer as A. Exactly. Both assertion and reason are true, and reason is the correct explanation. Obviously, why testis is situated outside the abdominal cavity? Because you need to maintain the temperature for this process of spermatogenesis, and for that your scrotal muscles are also helping you. Okay, to maintain the position of the testis. Next, now I will be talking about female reproductive system. Okay, so female reproductive system. The female reproductive system consists of a pair of ovaries along with a pair of oviducts, uterus, cervix, vagina, and the external genitalia located into the pelvic region. So here you can check this one pair of ovaries, a pair of ovaries, these are the primary reproductive organ of female. And if you talk about the secondary reproductive structure, there are one pair of oviducts, uterus, cervix, vagina, and the external genitalia located into the pelvic region. These are secondary reproductive organs of female reproductive system. Now if we talk about ovary as primary reproductive organ or as gonad or as primary reproductive organ, it can produce ovum by the process of oogenesis, and it can produce some female sex hormones called as estrogens and progesterone. These are obviously steroid hormone. These are also steroid kind of hormones and regarded as female sex hormone. Now, this part of the systems, now very important line written in NCERT, this part along with a pair of mammary glands are integrated structurally and functionally to support the process of ovulation, fertilization, pregnancy, birth and child care. So while we are talking about female reproductive system, why do we need to study about this mammary glands? As because you know that you will be talking about few hormones called as prolactin hormone, oxytocin hormone. Now these hormones or dopamine hormone, these hormones are having proper role uh, associated with these mammary glands, right? And these hormones, particularly prolactin, can influence the reproductive cycle as well. Now if a breastfeeding is going on, there will be formation of emotional bonding between the mother and the child, and that will give a positive feedback to mother's endocrine system to secrete more oxytocin and more prolactin, right? And if there is more prolactin or excess prolactin, after some time what will happen, that is gonna inhibit the follicle stimulating hormone, that's gonna inhibit the gonadotropin hormones. So that's why we need to talk about this mammary glands. And obviously without mammary glands, breastfeeding or baby feeding is not possible. It's a part of nurturing the baby. Now we'll talk about the anatomy of female reproductive system. Okay, so this diagram is already given in your NCERT book. So if we talk about female reproductive system, this part is the uterus. This part is the uterus. Now, at the junction of uterus and vagina, this tubular structure is the vagina. And at the junction of uterus and vagina, this narrow channel is called as cervical canal. Cervical canal or cervix, and together cervical canal and vagina will be forming the birth canal. Because during the time of parturition or child delivery, this passage will be followed. This passage will be followed. That's why this cervical canal and vagina together are forming birth canal. Okay. Now, the structure present ventral to the uterus and the reproductive system, that is the urinary bladder. And the structure which is present in front of this urinary bladder, this is the pubic symphysis, a cartilaginous structure. So, behind the pubic symphysis you got the urinary bladder, and behind the urinary bladder you got the uterus. Okay. Now this uterus is coming downwards, and here you will be finding the uterine. Here you are going to get the uterine opening or uterine meatus, and this vagina is coming out, and here you are going to get the vaginal opening. So this opening is called vaginal opening. Towards the dorsal side of our body, and towards the ventral side you are gonna get the uterine opening or urethral opening. From urinary bladder it's coming urethra, and urethral opening is here. So remember the position of urethral opening and vaginal opening. Vaginal opening is present towards the dorsal side of the body, and urethral opening for female is present towards the ventral side of your body. And both the opening, they are opened into this chamber kind of structure. This is called as vestibule. Both the opening, vaginal opening and urethral opening, they are open into this chamber called as vestibule, and this vestibule is guarded by few external genitalia kind of structure called as labia minora. Outside labia minora we will be getting labia majora, and outside labia majora you are gonna get mons pubis. Outside labia majora you are gonna get mons pubis, and these are some important structure of female external genitalia. And one more structure is observed towards the ventral end, which is actually exaggerated part of labia majora only. a small penis kind of structure observed here, that is called as clitoris. Okay. So clitoris, labia minora, labia majora, mons pubis, all of these are part of female external genitalia. So this is all about the basic anatomical structure of female reproductive system. Okay. So Ishika is asking, sir, what is the difference between cervix and vagina? Okay. So let me tell you, let me elaborate with another diagram. Okay. If you observe this particular diagram, this is the uterine cavity. This is the uterine cavity. Now we are observing from the front side. So this inverted pear-like structure you can observe here. This is the uterus or womb, and in this uterine cavity the development of baby will be going on. And this funnel-like, this tubular structure, this is the vagina, right? So this tubular structure, this is the vagina. Now the neck kind of, this narrow channel kind of region which is connecting vagina to uterine cavity, the narrow channel kind of region which is connecting vagina to uterine cavity, this part is called as cervix. Now you got the answer, Ishika. So cervix, basically, what is cervix? Cervix is nothing but lower part of uterus. Cervix is lower part of uterus, and through a channel called cervical canal, its uterine cavity is connected to vagina. So that's all about cervix, right? And this zone is very much crucial, this cervical region of female reproductive system. Right. So where were we? We were talking about the anatomy of female reproductive system. Okay. Now we'll be talking about ovaries, the prime female reproductive organs. Yes. So function of ovaries are written here. Female gamete formation means ovum formation and production of steroid hormones, estrogen and progesterone. The diameter, the length of ovaries are given as 2 to 3 centimeter length of each ovary, and ovaries are connected to pelvic wall with the help of uterus. Ovaries are connected to pelvic wall and uterus with the help of special fibrous structure called ligaments. Yes. So if this is the uterus, if this is the uterus and this is the vagina, this part will be the cervix region, this part will be the cervix region, and outside you are gonna get two ovaries, left ovary and right ovary. Left ovary, and right ovary. Now how these ovaries are attached with the uterine outer wall, or how these ovaries are attached with your abdominal cavity? So these particular structures are called as ligaments or ovarian ligaments. These ligaments are nothing but some fiber kind of structure which are holding tight this ovary with your abdominal region. Okay. Now we'll talk about histological. Now we'll be doing histological study of our ovary. So let's check that. So this is the outermost lining. So the outermost lining of ovary is called as ovarian surface epithelium. ovarian surface epithelium, the outermost covering of ovary, that is ovarian surface epithelium. Okay, just below to this ovarian surface epithelium, you will be finding one fibrous layer. Just below to this ovarian surface epithelium, you will be finding a fibrous layer that is called as tunica albuginea. Tunica albuginea. This is a similar kind of structure you got into the outer part or the outer covering of testis. Tunica albuginea is covering of ovaries as well as covering of testis. Okay, we haven't reached yet to the placenta, right, Manasha? We just have started the topic of female reproductive system. We are talking about ovaries. When I will be talking about placentation, gestation and development of fetus, then I will be giving you answer, right? So right now I'm discussing about the different layers of ovary. So the outermost layer is called ovarian surface epithelium. Below that we have tunica albuginea, right? And everything inside tunica albuginea is called as, this whole structure is called as ovarian stroma. This whole structure is called as ovarian stroma. Okay. Now, if you check the ovarian stroma, it's further divided into two portions, the inner ovarian stroma and the outer ovarian stroma. The outer ovarian stroma is called as cortex. Inner ovarian stroma is called as medulla. So ovarian stroma, how do you mean by ovarian stroma? The actual tissue present inside your ovary, that is called ovarian stroma, that is divided into two segments, the outer, the superficial part, that is called cortex, the inner part, that is called medulla. Now what is the importance of cortex and medulla? Prajwal is asking, sir, how two babies are born at once? Okay, the concept of twins, everything I will be discussing when I will be talking about fertilization, implantation and embryonic development. Okay, stay tuned. For that you have to stay tuned with this lecture. Okay, so what is the function of cortex region and medulla region? So in medullary region, you are observing that some blood vessels are present and also some nerves are present, nerve fibers are present. So basically, medullary region you will be finding blood vessels and nerves, and in cortex region what you can observe, developmental phases of the ovarian follicle and the ovum. So the process of ovum formation, the process of oogenesis or female gamete formation is taking place into the cortex region, and the entry of blood vessels and nerves are there through this into this medullary region. Medullary region is rich with blood vessels and nerves. Cortex region is actually providing the place for female gamete formation. So that is very basic things about the structure of ovary. Okay, now we will be talking about the structure of uterus and other associated structure. So here you can observe that both the side of uterus, as because this is the uterine structure or uterus, you can observe two ovaries are there. One is left ovary and another is right ovary, and every alternative month one of those two ovaries will be releasing one secondary oocyte, that is the immature form of the egg, right? So if this month your right ovary is releasing the secondary oocyte, the next month your left ovary gonna release another secondary oocyte, right? So in alternative months, right and left ovaries will be ovulating, that means releasing immature ovum in the form of secondary oocyte. This is called as ovulation. Okay, now this inverted pear-like structure is called as uterus, right? This inverted pear fruit kind of structure, and this upper part of uterus is called as uterine fundus. This upper part of uterus is called as uterine fundus. The cavity inside the uterus, that is called uterine cavity, and sometimes the uterus is also known as womb. What is the function of this uterine cavity? This will be giving place for the implantation and development of the baby and the embryo. The end part of the uterus, that is called as cervix, right? And inside the cervix a narrow channel is present, that is called as cervical canal, and outside that a tubular structure is present without any glands, and that is called vagina. What is the importance of vagina? During the time of sexual intercourse between a male and female, with the help of copulatory organ or penis, a male is going to inseminate or deposit the semen into this vagina zone. Remember, okay, and vagina is a non-glandular part. No gland is present inside this vagina. Okay, and what are the other structure, this layers, different layers of uterus? See, the innermost layer of uterus, that is called as endometrium. The innermost layer is called as endometrium. The middle layer of uterus, that is called as myometrium. Now by the name you can guess, myo means muscle. So myometrium is basically a smooth muscular structure made up of smooth muscle, and this myometrium is very much important during the time of childbirth or baby delivery. And the outermost layer of uterus is called as perimetrium, because it's covering the periphery. That's why the outer part is called as perimetrium. So these three are the layers of uterus: outside to inside, perimetrium, myometrium, endometrium; inside to outside, endometrium, myometrium, perimetrium. Now these two hanging hand kind of structure, we call this structure as a fallopian tube. We call this structure as fallopian tube. Another name of fallopian tube is called as oviduct. Now there are different portions you are gonna get in this fallopian tube. This funnel like structure is called as infundibulum. This broad and narrow structure, where generally sperm and ovum will exhibit fusion, means fertilization occurs. This zone is called as ampulla or ampulla. And this narrow part through which the fallopian tube is connected to uterine cavity, that is called as isthmus. Okay, now you can check here, some finger like projections are present. These are known as fimbri. These are known as fimbri. Fimbri, these are nothing but some finger like parts which are actually projection of infundibulum. Now if you check the infundibulum part, you will be getting very tiny opening, that is called as ostia, through which the ovum will enter into the fallopian tube. Okay, so from female reproductive system, from anatomy of female reproductive system, if you have from ovary, from uterus, if you have any question, you can write down in the comment section. I'll be happy to give you answer. Okay, so if we are done, we'll be moving forward. So the same thing is written as theory. So uterus is nothing but inverted pear shape of structure, kind of structure. Another name of uterus is called as womb. Uterus is attached to a pelvic wall with the help of some ligaments. It has three layers: perimetrium, the outer layer, there is a nothing but thin membranous layer; myometrium, the middle layer made up of smooth muscle; and endometrium, the innermost layer of uterus, that is made up of multiple glands. So that's why written as glandular layer. Now its myometrium is very much important. Its strong contraction during the childbirth will help or will aid the process of baby delivery, right? And the endometrium, this is also another important part, because when you talk about menstrual cycle, when you talk about implantation of the embryo, they are gonna take place, all these chaotic phenomena this during the time of menstrual cycle, they are gonna take place into this endometrium part. So whenever menstrual fluid is coming out, basically it's the rupturing, your body is rupturing some tissue and blood vessels, and they're happening inside this endometrium layer. Okay. Now we'll talk about external genitalia of female reproductive system. And what are these parts? These are called as mons pubis, labia majora, labia minora, hymen and clitoris. So if I draw the diagram, so this is the opening, this is the urethral meatus, this is the vaginal meatus, urethral opening, vaginal opening. Okay. This is the side towards the belly and this is the side towards the back side. Okay. So the outer fatty cushion kind of structure where pubic hair can be seen, this outermost part, that is called as mons pubis. Below mons pubis, fatty flap of skin can be seen, that is called as labia majora. Below labia majora, the skin is observed, that is called as labia minora. And at the ventral joining of labia minora, towards the ventral joining of joining side of labia minora, it will form a small penis kind of structure which is kind of homologous to male penis, and the structure is called as clitoris. The structure is called as clitoris. So, to remember this fact, this clitoris are formed by the joining of the ventral skin of labia minora. Okay, and this zone is called as vestibule. Towards the dorsal side, you will be finding vaginal opening. Towards the ventral side of our body, you will be finding urethral opening. So now let's check the theoretical part. So if you are asked about mons pubis, it's nothing but it's a fatty cushion kind of structure made up of fatty tissue and lot of pubic hair. If you are asked about labia majora, just below the mons pubis, you are going to get a fleshy fold. So in fatty cushion, mons pubis. It's fleshy fold, it's labia majora. Now below labia majora, you're gonna get a paired fold under labia majora. They are called as labia minora. Labia minora are originated from labia majora skin, and from labia minora, clitoris will be originating. So it's a finger-like structure at the upper junction of labia minora skin. And one more important structure you need to talk about here, that is hymen. So what is hymen? It's a membranous structure that partially covers the vaginal opening, a membranous structure that will be partially covering the vaginal opening, and that covering is called as hymen. And often it was said that it was actually it was a myth. During the time of first coitus, this hymen will be ruptured and there will be bleeding, but that myth is busted nowadays, right? Okay, so we are done with external genitalia of female reproductive system. Now the myth buster is here. So the hymen is often torn during the time of first coitus, that is called sexual intercourse. However, it can also be broken by a sudden fall or jolt, a sudden shock of your body, insertion of vaginal tampon, like few female will be using this vaginal tampon, cottony pad kind of structure, which are inserted into the vagina to absorb excessive menstrual fluid. Or if you are actively participating in sports like horseback riding, cycling, swimming, then also simply this hymen can be torn or can get ruptured. Yes, in some women the hymen persists even after coitus, right? So that's why the fact is that the presence or absence of hymen is not a reliable indicator for virginity or sexual experience. So in some old days people used to talk about if a female, if a woman is a virgin, then there should be intact hymen present just after marriage. They used to check like in this manner. But as because we are studying science, we are studying anatomy and morphology of human, so it's quite clear. So this is not a fact at all, right? Okay. Now we'll talk about female, the last part of female reproductive system, that is mammary gland. Though these glands are not associated with directly with female reproductive system, but the functionality can influence the reproductive cycle. Okay. Now when you talk about mammary gland, they are exclusive feature of mammals, right? And you think that only mothers, only females have these mammary glands, but mammary glands are present in all kinds of mammals, right? In male as well as in female. In case of female, mammary glands are functional during the time of motherhood, and in case of male, mammary glands are vestigial or non-functional. Okay. Now Ishika is saying, is there any method to check in men's? No, as I said that for female also it cannot be checked like either she is virgin or not, and obviously for men also it cannot be checked either he is virgin. Okay. So all those talk are worthless or baseless or wax kind of talk. So scientifically those things are not possible. Okay. Okay. So I was talking about mammary gland. So I said that mammary gland is present in case of male as well as female. For female, mammary glands are functional, and for males, mammary glands are non-functional or vestigial in nature. Okay. And one more interesting fact about mammary gland, they are nothing but they are modified sweat gland. Mammary glands are modified sweat gland. Mammary glands are modified sweat glands. Okay. Now if you check inside one mammary gland, you are gonna get 15 to 20 chambers called as mammary lobes. Each mammary gland is having 15 to 20 mammary lobes, and inside each mammary lobe you are gonna get some milk producing structure called as mammary alveoli. Mammary alveoli, these are the milk producing structure where milk is formed under the influence of prolactin hormone. Remember that? So, inside mammary lobes, you are gonna get mammary alveoli, and from mammary alveoli, the duct is coming out, that they are called as mammary tubules. Multiple mammary tubules will be joining together to form mammary duct. Multiple mammary ducts will be joining together to form mammary ampulla or ampulla, and this mammary ampulla will come out through lactiferous duct, and lactiferous duct ultimately open through nipple. Okay, so this chronology you need to study or understand it conceptually. The passage of milk secretion, again I am repeating it, mammary gland is nothing but modified sweat gland. Okay, one mammary gland is having 15 to 20 chambers. Every mammary gland is having 15 to 20 chambers inside it, right? And they are called as mammary lobes. Inside mammary lobes, you will get multiple milk producing structures, and they are called mammary alveoli. And this mammary alveoli are under control of which hormone? Prolactin hormone. Milk producing hormone is called prolactin, right? Now, under influence of oxytocin from mammary alveoli, milk will be coming out, right? Milk secretion is influenced by oxytocin hormone, milk production is influenced by prolactin hormone. Okay, now what is there after mammary alveoli? Multiple mammary alveoli will be joining together to form mammary tubules. Multiple mammary tubules will be joining together to form mammary duct. Multiple mammary duct will be joining together to form ampulla or mammary ampulla. Ampulla will be coming out through lactiferous duct, and ultimately milk is gonna come out through nipple. Okay. Now, surrounding this nipple region, you are gonna get a dark zone. Surrounding the nipple region, you are gonna get a dark zone, and that dark zone, that dark area of mammary gland is called as areola. Okay, so that's all about mammary gland. Now let's moving forward to the next topic, that is called as gametogenesis. Okay. Ishika is asking, what is difference between alveoli, tubule or duct? Let me tell you. Okay, let me draw another diagram. So if this is the mammary gland, and that is divided into many chambers. Suppose these are the different chambers of mammary gland. Now inside you are gonna get multiple balloon-like structure. Inside these balloons, milk formation is going on. So these balloons are called as mammary alveoli. These balloons are called as mammary alveoli, inside which milk formation is going on, right? Now, the duct from each balloon will come out. So these ducts which are coming out from alveoli, we call them what? We call them mammary tubule. We call them mammary tubule, right? Now this mammary tubules of many mammary tubules are coming together, and they are forming a common passage, common tube, that is called as mammary duct. And multiple mammary duct will be joining together to form ampulla. And from ampulla the tube is coming out, that is called lactiferous duct, and from lactiferous duct, the opening is through the nipple region. So that is the difference between alveoli, tubule or mammary duct. So inside mammary alveoli, milk formation is going on. Then milk is gonna come out through some small tube kind of structure. We'll be calling them as mammary tubules. Multiple mammary tubules are gonna merge together to form large tube, that is called as mammary duct, and multiple mammary duct gonna merge together to form a bulged portion, that is called as mammary ampulla, and ampulla will be throwing out the milk or eject out the milk with the help of a channel or tunnel, that is called as lactiferous duct, and the opening of lactiferous duct is nipple. Now gametogenesis. Okay, so gametogenesis means the process of formation of sperm and ovum into gonads. Okay, first we'll be talking about spermatogenesis. And already I have told or I have taught in this lecture that which is the place inside male reproductive system. Exactly you need to answer me where this spermatogenesis is taking place. Can you write down in the comment section? The spermatogenesis takes place into which part of testis? You have to write down in the comment section. Spermatogenesis takes place into which part of testis? So the answer is spermatogenesis takes place inside seminiferous tubules. You need to remember this information. Yes, seminiferous tubules. Okay. So inside seminiferous tubules, you are finding some cuboidal or ovoid cuboidal shaped cells. These are called as spermatogonia. These are called as spermatogonia. Now spermatogonia, these are diploid cells. Spermatogonia, these are nothing but diploid cell having 46 chromosomes into them. Spermatogonia. Now spermatogonia will perform mitosis, and they will form multiple similar kind of cells or many spermatogonial cells. So basically here I need to write it as spermatogonium. One spermatogonium will divide by the process of mitosis and will form spermatogonia. Remember, these spermatogonia, these are diploid cells. Okay. Now what will happen inside the seminiferous tubule? Yes, Ishika, I have seen your answer. That's you are doing quite good. Okay. So inside seminiferous tubule, now this spermatogonia will change the shape, will start to showing enlargement. It will gain mass. It will become a larger cell, right? So that process is called as differentiation. The process is called as differentiation. Now this spermatogonia will differentiate . Remember, differentiation is not a kind of cell division, it's just that cell is changing the shape. That is called differentiation. Now, the spermatogonia will undergo differentiation and it will make a large cell called as primary spermatocyte. It's called as primary spermatocyte. Now, as because there was no cell division, primary spermatocyte also containing 46 chromosome or 2n number of chromosome. Okay. Now, from here the real story begins. What happened? This primary spermatocyte will start another kind of cell division that we call as meiosis. Right. Now, this primary spermatocyte will start undergoing first meiotic division. I am writing it as M1, first meiotic division, and we know that this first meiotic division is kind of reductional division. Reductional division. Do you know what is the difference between mitosis and meiosis? Mitosis, by the process of mitosis, if cell division is going on, then a mother cell will produce two daughter cell with similar number of chromosome. But if a mother cell is performing meiosis cell division, then it will be producing two daughter cell but with half of the number of chromosome. So, for mitotic division, new cells will also contain 2n number of chromosome, but for meiosis division, new cells will be containing n number of chromosome. So, primary spermatocyte will undergo M1, that means first meiotic division, that is a reductional division, and the resultant cells are produced here. Resultant cells are produced here. We will be calling them as secondary spermatocyte. Secondary spermatocyte. And as because they are resultant cells of meiosis 1, the chromosome number will be n, the chromosome number will be 23, half, directly half. Now, the secondary spermatocytes will undergo meiosis 2 or second meiotic division, right? The secondary spermatocyte will undergo meiosis 2 or second meiotic division, and the resultant cells are produced here. Now, we call them spermatids. Spermatids. Now, if you question yourself that how many chromosomes are present into each spermatid, now as because meiosis 2 is kind of equational division, so there is no change of chromosomes. So these spermatids also have haploid set, means 23 chromosomes only inside them, right? Now, from spermatid there will be another set of differentiation, means the spermatids gonna change their shape now. There will be no cell division, there will be only differentiation. The spermatids gonna change their shape now, and the spermatids, we will call them, they are looking like tadpole, they have flagella, we call them spermatozoa or sperm. Simply you can call them sperm, and they have also haploid, means 23 numbers of chromosomes, right? So, this is the whole sequence of spermatogenesis, and the question is asked in the way that one primary spermatocyte can produce how many secondary spermatocyte? One primary spermatocyte can produce two secondary spermatocyte, and one primary spermatocyte can produce four spermatid and four sperm. Why it's like that? If this is the primary spermatocyte, this will undergo what? This will undergo meiosis 1 and will produce two secondary spermatocyte. And remember, secondary spermatocytes are the first haploid cell of spermatogenesis process. Secondary spermatocytes are the first haploid cells in the process of spermatogenesis, and each secondary spermatocyte will undergo meiosis 2. Each secondary spermatocyte will undergo meiosis 2 or second meiotic division and will produce total 4 cells, right? And we call them spermatid. We call them spermatid. They also have haploid set of chromosome, and one spermatid will get converted into one sperm. So that's why this calculation you have to remember. One primary spermatocyte is giving rise to secondary spermatocyte, and 4 spermatid and 4 spermatozoa. So it's as simple as that. Now, these 3 different terminologies are quite confusing for many of the students: spermatogenesis, spermiogenesis, and spermiation. Now, what do you mean by spermatogenesis? The total process of sperm formation starting from spermatogonia to spermatozoa. This whole process is called as spermatogenesis, but only the last step where spermatid to spermatozoa formation is happening, this process is called as spermiogenesis, right? So what is spermatogenesis? Spermatogenesis means from spermatogonia through multiple stages formation of sperms. So this process is called as spermatogenesis. What is spermiogenesis? The last stage of spermatogenesis where from spermatid, by the process of differentiation, sperm is formed. That is called spermiogenesis. And what is spermiation? Now, spermiation is a quite different phenomena. We know about this Sertoli cells or this nurse cell, right? So when sperms are formed newly, then we'll be finding they are attached with the Sertoli cells. Which cell is this? This is Sertoli cell. Newly formed sperms are initially, you'll be finding they are attached with Sertoli cell. Now, when your body will feel that, yes, these sperms are matured now. When these sperms are structurally matured, then what will happen? The Sertoli cell will free them. Up. Now they will move further inside the seminiferous tubules, and this process of releasing of structurally matured spermatozoa and the detachment with Sertoli cell, this process is called as spermiation, the release of sperm from seminiferous tubules. Okay. Now, if you have any question from the process of spermatogenesis, you can write down into the comment section. So this is the passage of sperm. Gradually it's coming out. See, the sperm is formed inside the seminiferous tubule. From seminiferous tubule, sperm will come out into the retitestis region. From retitestis, it will come into the vasa efferentia. From vasa efferentia, it is it will be coming into the highly coiled zone, that is called epididymis. From epididymis it will come into the vas deferens. Now you know that vas deferens will join together with seminal duct and will form ejaculatory duct, and this ejaculatory duct will join with urethra. The ejaculatory duct will join with prostatic duct or prostatic urethra and will be forming urinogenital duct and will be released out through the urethral orifice of penis during the time of insemination. During the time of insemination, the sperms will be released out through urethral orifice of penis. So this is the passage of sperm secretion, or sorry, the insemination of sperm. Okay, from 4 o'clock the session is running. Okay, now let's come to next topic, that is the structure of structurally matured spermatozoa. Okay, so this is structure of mature spermatozoa, and spermatozoa is divided into three part. First, four part. First head, then neck, second part, third part is the middle piece, and the fourth part, that is tail. So a structurally matured sperm is having four distinct parts: head, neck, middle piece and tail. Okay. Now if you observe the head region, the cap portion of the head, you will be finding a special structure filled with enzymes or hydrolytic enzymes, which will be helping the sperm during the time of penetration or during the time of fusion with the ovum, during the time of fertilization. The enzymes secreted by acrosome, we call them sperm lysine. Sperm lysins, and you know that this acrosome is nothing but modified Golgi complex or modified Golgi body. Okay. Now beneath the acrosomal part, we will see a condensed nucleus, and this nucleus is haploid in nature, that is having only 23 chromosome. Okay, so that's all about head part. Now let's come to the neck region. Now the neck region is having two cell organelles called we call them centrioles. So the centriole which is situated towards the nucleus, that is called as proximal centriole. The centriole which is situated towards the middle piece part, that is called distal centriole. The centriole which is present below to the proximal centriole, that is called distal centriole. Now both these centrioles have different functions. Distal centriole is responsible for the formation of this axial filament or this flagella of the tail, and during the time of fertilization, sperms will be donating the nucleus as well as proximal centriole to the ovum. Okay. Sir, which is the smallest cell in human body? Obviously sperm, right? If you consider the head, the size is only 5 micrometer, 5 micron. So that's why we consider sperm as the smallest cell of human body, right? Okay. I was talking about function of proximal and distal centriole. So it's very easy. Distal centriole is responsible for formation of this tail, and proximal centriole will be donated to the ovum during the time of fertilization. Okay, now it's time to check the middle piece zone. So into the middle piece, what you are observing here, a spiral of mitochondrial bunch, right? So this spiral structure is nothing but hundreds of mitochondria present into the middle piece zone, and you know that when a sperm is swimming, it will be utilizing fructose as the energy material, and this mitochondria, they will be responsible for production of energy for sperm swimming, and with the help of the tail, by the propulsion of the tail, sperm can move forward. So the function of tail is swimming, and the function of mitochondria is to provide energy source during the time of swimming. So that was all about the structure of sperm. Now if you want to ask any question, you are free to write down into the comment section. I will be happy to answer. You can ask any kind of conceptual question, whatever the topics we have covered till here. Okay. Okay, so if you don't have a question, I'll be moving forward to the next part. So while we are studying about this whole process of spermatogenesis, uh, average human male ejaculates near about 200 to 300 million of sperms during a coitus, right? Basically what happens during the time of one coitus, a male releases 2 to 4 ml of semen, right? What is semen? It's containing the liquid medium as well as the sperms are thriving inside it. Okay. So Prajal is asking, sir, what is the viability of sperm? Viability of sperm means outside the male body upon ejaculation, it can survive for 48 hours, right? So that is the viability of sperm. Okay. So a mature adult male can ejaculate 2 to 4 ml of semen, and that 2 to 4 ml of semen is containing near about 200 to 300 millions of sperms into it, right? But for normal fertility, at least 60% of these 200 to 300 millions of sperms must have normal shape and size, and at least 40% of them must be showing vigorous motility. What do you mean by vigorous motility? To move very fast by the whiplash movement of the tail or flagella. That is called vigorous motility, the capability of swimming very fast. Yes, Niharika, it's 60% and 40%. Okay. Uh, Now, this is very contradictory actually, what NCERT is trying to say. Okay. In many books of higher medicine, they are saying it should be 60% of total sperm count, and then you have to calculate the 40% of this 60%. But many doctors, many good doctors are saying it should not be like that. It should be 60% should have normal shape, 60% we have to calculate for this 200 to 300 million, and this 40% the sperms with vigorous motility, we have to calculate in terms of this total sperm count. Okay. So that's making the topic too much confusing, because in higher medicine, whenever you are studying some books, right, like if you talk about the Guyton and Hall, the the Bible for medical physiology, okay, there you can opt for this kind of explanation. Okay. Now It's written here, sperms released from the seminiferous tubules are transported by the accessory ducts, and secretion of epididymis, vas deferens, seminal vesicle and prostate are essential for maturation and motility of sperm. So this seminal plasma along with the sperm constitute the semen. Now, one more important thing I am telling here, remember this. You can be asked, first thing, about the structural maturity of sperm. And second thing can be asked as functional maturity. Structural maturity of sperm and functional maturity of sperm. What are the places, okay, where sperms are getting the structural maturity? So this is very easy to answer. They are seminiferous tubule. But freshly produced sperm inside the seminiferous tubule, which are coming out from seminiferous tubule, they cannot swim, right? So that's why we cannot say those sperms are functionally matured. Sperms are functionally matured from that moment when a sperm can swim on its own. So for the first time we can observe swimming capability of sperm inside epididymis. So epididymis is the place for the functional maturity of sperm, and seminiferous tubule is the place for structural maturity of sperm. Remember this. Okay. Now we were talking about semen, right? So semen is nothing but sperm plus seminal plasma. Sperm plus seminal plasma. We all know about that ejaculum is having 2 to 300 millions of sperm, and the seminal plasma is rich with fructose, calcium, enzymes, citric acid. Okay, so this is about seminal plasma, and you know that seminal plasma is nothing but secretion of those glands called seminal vesicle, prostate, and bulbourethral gland. Okay, now we'll talk about oogenesis, the female gametogenesis. So the process of formation of a mature female gamete is called oogenesis, which is marked by markedly different from spermatogenesis. Why it's so? Because the process of oogenesis will be the initiation of oogenesis is during embryonic life. During embryonic life, when the girl child is as fetus gradually growing inside the mother's womb, and her age is near about 20 week, right? So nearby 20th week of the fetus developmental phase, inside the ovary of the developing girl child, oogenesis process will start. Okay. Now what are the sequential stages of oogenesis? Like first you can observe formation of oogonia into ovaries. Upon successful division of oogonia, you can observe formation of primary oocyte, the next phase of oogonia. Upon successful division of primary oocyte, you can observe formation of secondary oocyte, and if by chance there is fertilization, then only secondary oocyte gonna complete the next phase of cell division to produce ootid, and this ootid is nothing but the mature ovum, right? So these are the different phases of oogenesis, right? And one more thing you need to remember, no oogonia formed or added after birth. Why it's so? It's a line, very important line of NCERT. This oogonia, these are formed during the time of embryonic phase, and all the oogonia will be converted into primary oocyte again during the time of embryonic phase. So, after birth or after puberty, during the time of puberty or after birth, if you are checking inside the female ovaries, you are not gonna get oogonia, rather you are getting the primary oocyte stage of maturing ovum. So that's why NCERT is saying that no oogonia formed or added after birth, because oogonia is the first step, and already during the time of the 20th week or of embryogenesis, all of this oogonia will be converted into primary oocyte, right? Okay. Now, one more thing is written here, at puberty only 60,000 to 80,000 primary follicles are left into each ovary. Yes, that's a true fact. So, if you observe during the time of embryogenesis before birth, total you will be finding millions, 2 to 4 millions of primary oocytes are formed from oogonia before birth. But after birth, what happens? Gradually inside the ovary of female, the primary oocytes along with the primary follicles will be dying gradually. That is called atresia phase, death of primary oocyte, and only 60,000 to 80,000 primary follicle or primary oocytes gonna survive till the age of 12 years or 13 years. Okay. Now let's talk about different phases of this oogenesis. Okay. Now to understand oogenesis in a vivid manner, in a detailed manner, we need to understand some structures present into the cortex region of the ovary, right? See, uh, so when this oogonia are getting converted into primary oocyte, they will be encaged. They will be encaged by special structure. We call them primary follicle. So inside primary follicle, the immature form of the egg you are gonna get, that is primary oocyte. 1 degree means primary. So inside primary follicle, you are gonna get this primary oocyte, right? Now, what during the time of puberty at the age of 12 to 13 years, what is gonna happen? The primary follicle will start maturation. The primary follicle will start maturation, and this primary follicle will get converted into secondary follicle. Primary follicle will get converted into secondary follicle. Now, if you check inside the secondary follicle, still the phase of the ovum or the egg you will be getting, that is primary oocyte. It's not changed yet. But once the follicle is changed into secondary follicle and it's going towards tertiary follicle, now what will happen? The primary oocyte will complete the division, and inside the tertiary follicle you will be finding it has completed the division and to form secondary primary oocyte to secondary oocyte. Now, many people are getting confused. Let me clarify again. What are these follicles? Follicles are kind of cage, kind of football kind of structure which are encaging the maturing egg or maturing ovum. So, the cage is called follicle, and inside the cage you got a bird, that is the immaturing phases of ovum, that is your oocyte. So, inside primary follicle you got primary oocyte, inside secondary follicle you got primary oocyte, but when the follicle is matured into tertiary follicle stage, then inside the tertiary follicle you are gonna get secondary oocyte. Remember this. Now gradually the tertiary follicle will grow and it will form a larger version of tertiary follicle. Now we say that the follicle is matured, and that is called Graafian follicle. Even inside the Graafian follicle, the stage of egg you are gonna get that secondary oocyte, 2-degree oocyte. And you know that Graafian follicle is the mature follicle. So what is gonna be happen after this? The ovary will throw out the secondary oocyte. The ovary will throw out the secondary oocyte outside, and this process is called as ovulation. This process is called as ovulation. So you got a question here, you got an important question here: which phase of maturing egg is ovulated? So it's not the ovum, it's secondary oocyte, right? Now once this Graafian follicle is kicking out the secondary oocyte outside the ovary, within few hours the Graafian follicle will get converted into yellowish structure called as corpus luteum. Once the Graafian follicle is ovulated, thrown out the secondary oocyte, it will get converted into yellowish structure called as corpus luteum. Now this corpus luteum will be acting as a temporary endocrine gland inside the ovary. This corpus luteum will be secreting progesterone, the corpus luteum will be secreting estrogen. And why it's looking yellow in color? Because it's containing some yellow pigment called lutein. That's why we call it corpus luteum. But within few days, the corpus luteum also will die, and it will make a white colored scar while the corpus luteum is dying. So the dying corpus luteum, which is white in color, that is called as corpus albicans. Luteum, corpus luteum. Luteum means yellowish body and albicans means whitish body. So these are the different structures of follicles you are gonna get inside the ovarian cortex, right? It's starting from primary follicle to secondary follicle to tertiary follicle to Graafian follicle. Now this is called the mature follicle. Now after Graafian follicle there will be ovulation. Now that empty follicle will change itself to a yellow colored body, that is called as corpus luteum. And the corpus luteum, when it's dying within 10 to 11 days or 12 days, it will become pale yellow or whitish in color, and it will shrink gradually by forming corpus albicans. Okay. Now if you have any question from this particular topic, you can write down your comment. Okay. So Asika, what is causing the ovary to rupture and release the secondary oocyte? See, focus carefully. Focus over here into the Graafian follicle. So this enlarged follicle, so the largest follicle is called Graafian follicle, and inside this Graafian follicle you will be finding a liquid filled pool that is called as antrum. Though the antrum was formed during the time of tertiary follicle. So this liquid filled pool is called as antrum, right? So this antrum is filled with some liquid. Now what happened? Once this Graafian follicle is formed, some hormone will be influencing like anything. So what are those hormones? It's LH, luteinizing hormone mainly, right? And along with luteinizing hormone, FSH will be influencing it. Along with FSH, estrogen will be influencing it. So all three hormones together gonna influence this Graafian follicle, and now what will happen? The cells inside the Graafian follicle will start leaking, start secreting more liquid called as liquor folliculi, the pool, the liquid pool inside the antrum. So what will happen? If you are pouring water inside a balloon, so after a certain time the balloon will get blasted, right? The same thing happens here. So once the antrum is totally filled with liquid, so the pressure caused by the liquid inside it, it will create a huge force, and the Graafian follicle will blast, and that blast will throw out the secondary oocyte out the ovary. You got your answer, Ishika? Okay. Niharika has asked a question. Sir, what are the functions of corpus luteum? As I said, beta ki corpus luteum is acting as a temporary endocrine gland. Corpus luteum is acting as a temporary endocrine gland. Now, what happens? This corpus luteum is under control of luteinizing hormone, right? Corpus luteum is under control of luteinizing hormone, and when it's matured, when the corpus luteum is matured, it will start production of progesterone, the prime hormone produced by corpus luteum. Corpus luteum also can produce very good amount of estrogen, and during the time of pregnancy, this corpus luteum can produce a special hormone called as relaxin, and another, the fourth hormone produced by corpus luteum, that is called as inhibin. Right. Now, if you ask me, sir, what is the function of progesterone? Progesterone is required for to maintain the pregnancy. If that female is pregnant, to retain the pregnancy for 9 months, you need high amount or high volume, high titer, high concentration of progesterone into mother blood. Estrogen is required to make the uterine wall thick. If you are unable to make the uterine wall thick, then there will be no implantation, no pregnancy will be there, right? Relaxin is required during the time of childbirth or parturition. So that is the importance of corpus luteum. Okay, but what happens in normal time, in general months, the corpus luteum will appear and it will die within 10 to 12 days, as because there is no fertilization, no pregnancy is occurring into the female body. Yes, Suhaib, I think you are Sania, right? I remember your name. Very good evening. Okay, so this is the diagram I was talking about. Here I have kept all the forms of the follicles present inside the cortex of ovary. So here you can see the 1 degree F means it's primary follicle, and here inside the primary follicle the egg cell is there, that is primary oocyte, and the cells which are surrounding the primary oocyte, they are called as follicular cells. Okay. Now this follicular cells means the normal cells which are surrounding the ovum, they are also called as granulosa cells. They are also called as granulosa cells. Now if you talk about the advanced version of primary follicle, it's secondary follicle, and the phase of the egg you are going to get, that is similar, primary oocyte only. Now, what's the difference between secondary follicle and primary follicle? You will be finding there is more cells or more layers of this granulosa cell or follicular cells which are surrounding the primary oocyte, and one outermost covering, that is called theca. The outermost covering of secondary follicle is called as theca formation. Now, advanced version of secondary follicle is called as tertiary follicle. It's even larger. Okay, so inside tertiary follicle, the phase of egg you can observe, that is secondary oocyte now. 2-degree oocyte means secondary oocyte now. There are more number of granulosa cell surrounding the secondary oocyte, and you will find one pool filled of liquid that is called as antrum. So according to NCERT, for the first time the antrum can be seen inside tertiary follicle or 3-degree follicle, and this theca is differentiated into outer theca externa and inner theca interna. Two layers of theca can be observed in tertiary follicle, but in case of secondary follicle there was only one theca. Okay, now advanced version of tertiary follicle, that is the mature follicle or Graafian follicle. Inside Graafian follicle, what you can observe, the similar phase of the egg, that is the secondary oocyte can be seen. More number of follicular cells are there, but the antrum is largest now. Antrum is largest now along with theca externa and theca interna, and this Graafian follicle will do the phase of ovulation under the influence of LH hormone along with progesterone, along with FSH and estrogen. And after ovulation, the Graafian follicle will change itself to a yellow colored body called corpus luteum. The corpus luteum in normal days, normal condition, it will sustain for 10 to 12 days, then it will die to form a white colored body, that is called corpus albicans. Remember, corpus albicans is white in color, corpus luteum is yellow in color. Okay. So I got another question from Ishika. Sir, if estrogen is high and progesterone is low, then what will happen? At what time? During the time of pregnancy or in normal days? When? Estrogen is high and progesterone is low during the normal time, normal menstrual month, or during the time of pregnancy? I didn't get you, Ishika. Can you please rewrite your question explaining it? So if you're talking about normal menstrual month, so obviously in normal time estrogen is high and progesterone is low, except if you consider the luteal phase. Okay, bye bye Sania. Okay, so you are asking about pregnancy, during pregnancy. Okay, so what will happen then? Simply abortion will take place, spontaneous abortion, right? uh, for during the time of throughout the course of pregnancy, you need to keep high titer or high concentration of estrogen as well as progesterone, right? But if there is high estrogen but low progesterone, then it won't work. That's why you know that progesterone has got another name called as pregnancy hormone. As soon as the progesterone will fall, the uterine endometrium will stop getting blood supply, and if blood supply is disturbed, then what will happen? Ultimately the placenta will shrink, the embryo will run out of nutrition and it will die, and there will be a spontaneous abortion or spontaneous uh, termination of pregnancy, right? But yes, remember this thing, but just prior to the parturition, just prior to the childbirth, it's mandatory to keep estrogen high and to put or to drag the progesterone into a low level. Otherwise, there will be problem during the time of childbirth or baby delivery. So throughout the pregnancy, you need high progesterone, high estrogen, but during the time of parturition, you need to keep the estrogen high, but you need to drag the progesterone into very low level. And the reason is during the time of parturition, you will be needing oxytocin, right? So if there is high progesterone, oxytocin cannot work in a proper way, remember. Okay, so now let's check the process of oogenesis in detail. So we are talking about embryonic life. So during the time of embryonic life, inside the ovaries you can observe some diploid cells called oogonia, right? Now, during the time of embryonic life only, this oogonia will perform mitosis and differentiation, to form a slightly different kind of cell called as primary oocyte. So it's during the time of embryonic life, oogonia will be forming primary oocyte, and primary oocyte are the resultant cell of mitosis and differentiation. Now what will happen? This primary oocyte will start. The primary oocyte will start first meiotic division. The primary oocyte will start first meiotic division, but you know, this primary oocyte, they are surrounded by these follicular cells or granulosa cell. They will secrete some chemical inhibitor, and suddenly what will happen? The meiosis 1 will be arrested at prophase 1 phase. So at prophase 1 phase, prophase 1 of meiosis 1, the division, first meiotic division will be arrested during the time of embryogenesis, because that's quite harmful for the development of the baby, right? So that's why the process will be arrested. Now within some days, within few months there will be birth, and after birth within 11 to 13 years it will arrive the puberty. Okay, now what will happen? During the time of puberty, the female ovary will get influence or get stimulus by FSH and LH. The female ovary will get stimulus from these two hormones, FSH and LH, during the time of puberty. So that will resume the incomplete meiosis 1 now, and now what will happen? Meiosis 1 will be completed. Meiosis 1 will be completed after puberty under influence of follicle stimulating hormone and luteinizing hormone. Now once meiosis 1 is completed, this primary oocyte will form two cells. One is large cell called as secondary oocyte, and one is kind of very small cell called as first polar body. And remember, this primary oocyte was a diploid cell, but the resultant cells, that means one first polar body and one secondary oocyte, they are haploid in nature. So if you are asked that which is the first haploid cell in oogenesis process, so they are first polar body and secondary oocyte. Now in course of division, the first polar body will die. It cannot sustain for a long time, but the secondary oocyte will be ovulated or thrown out of the ovary. You know the process of ovulation, right? Now if it's not fertilizing the sperm, okay, now if it's not fertilizing the sperm, this secondary oocyte, then it will die as secondary oocyte. But if sperm is arising and fertilization is successfully going on, then what will happen? Upon successful fertilization, the secondary oocyte will complete second meiotic division, and will form two new cell. One is the mature ovum or the oocyte, another the smaller cell called second polar body. Now this oocyte or ovum, that is also haploid, second polar body, that is also haploid in nature. Now we know that during the time of cell division, either it's mitosis or meiosis, generally one mother cell will give rise to daughter cell, and they are of similar size. But why during the time of this oogenesis, the primary oocyte will give rise one large cell and one small cell? Can you give the answer? What is the reason behind this? Normally, if a cell divides to new progeny cells or to new daughter cells, they will get similar size and shape. But why it's so that during the time of oogenesis process, if when the primary oocyte is dividing into secondary oocyte and first polar body, secondary oocyte is large, first polar body is small, and the secondary oocyte is dividing, the oocyte is large, second polar body is small. So why there is uneven cytokinesis is happening? We call it equal karyokinesis but unequal cytokinesis. So why it happens during the time of oogenesis? Can somebody answer this question? Write down into the comment section. No, a reductional division, obviously. Primary oocyte, it was diploid, but secondary oocyte will be haploid, polar body will be haploid. So like this will be the condition of the distribution of the chromosome. Okay, but my question is why one cell is bigger and one cell is smaller? Why one cell is getting almost all the cytoplasm and another cell is not getting cytoplasm? Why? There is equal nuclear division but unequal cytoplasmic division. Why it's so? Can you answer that? In normal mitosis, mitosis took place during the time of spermatogenesis as well, but in that case there was no unequal division, right? But why there is unequal division? One cell large, one cell small. My question is that. Okay. Is there anybody who knows the answer? Okay. I'm telling the answer. Remember, when this oogonia was divided into primary oocyte, and the primary oocyte had started meiosis 1 or first meiotic division, right? No, Niharika, that's also not the answer. When the primary oocyte started the meiosis 1, but it got arrested, right? And what do you think, for how many years it remained arrested? For how many years it remained arrested? Near about 11 to 13 years. If you calculate from the embryonic stage, then within few months there will be birth, then from birth to puberty it's 11 to 13 years. So it's almost 13 years it remained arrested, and then when arrest is revoked, it continues meiosis 1 and completed and formed secondary oocyte and first polar body. During the long span, it has lost one of the crucial cell organelle which is required for animal cell division. Can you name that? Can you name that? Which cell organelle is very much crucial during the time of animal cell division, which is very much crucial for cytokinesis? Is there anyone else? Without which cell organelle is animal cells cannot divide? Generally, we know the answer, right? Our neurons do not have that cell organelle. Now, can you guess that? Okay, I'm telling the answer. It's centriole. Yeah, centrosome. Ishika, you guessed it somehow correct. Centrosome, rather we say it centrioles nowadays, right? So uh, during this span of 13 years of wait, 13 years of waiting. Hello, very good evening, Chaitanya. That primary oocyte will be losing the centriole, and that is the reason that centriole is responsible for formation of the astral rays and the spindle, and those spindle will be helping the cell to determine the equal cytoplasmic division. So, as because the primary oocyte will be losing the centriole during the time of completion of meiosis 1, it cannot determine to divide the cytoplasmic material equally. That is why one cell will get almost 90% of the cytoplasm, that will become secondary oocyte, and that will reserve a lot of resource or energy for future, and another cell almost will get nothing or except this nucleus. So, that is the reason it lost the centriole. Okay. Now, we'll study about the structure of secondary oocyte. We will talk about the structure we have already studied the structure of sperm. I will talk about structure of not ovum but secondary oocyte, because when it's ovulated from the ovary, it's not the ovum, it's secondary oocyte. See, this is the nucleus, and we know that nucleus is already haploid in nature, having 23 chromosomes. This is the cytoplasm, and the cytoplasm of ovum is called as ooplasm. Ooplasm. Okay. Now, inside this cytoplasm, we'll be finding some vesicles filled with some glycoprotein molecules. So, these vesicles are called as cortical granules. Some special structure you'll be finding into egg, right? Okay. Now, this is the cell membrane of ovum, a cell membrane of secondary oocyte, and cell membrane of secondary oocyte is called as vitelline membrane, a very much crucial membrane for ovum or egg. Okay. Now, what is covering the vitelline membrane? The outer covering of vitelline membrane is called as zona pellucida, a very important structure of egg. Zona pellucida, which is present outside the cell membrane of egg or outside the vitelline membrane. Now, the space in between zona pellucida and vitelline membrane is called as perivitelline space. Now, what is the importance of this zona pellucida? You know, if you take a test tube and if you take sperm of monkey and sperm of human, an ovum of human being, so what do you think? What will happen? Will the sperm of monkey can fertilize ovum of human? Can you answer? If in laboratory, in artificial condition, if you are taking sperm of monkey and ovum of human being, so what do you think? Will they be fertilizing each other? You can write down your comment. Or if you take ovum of dog and sperm from a fish, will they be fertilizing each other? No. Yes, they won't be fertilizing each other. That is called species. That is called species specificity. Only sperm of a certain species can fertilize ovum of that particular species only, right? Yes. So, the species specificity is determined by the zona pellucida of ovum. So, zona pellucida is having some lock kind of structure, and if it's human sperm, then only you can open the zona pellucida lock with the help of key. So zona pellucida is responsible for species specificity. What is species specificity? Only human sperm can fertilize human ovum. Only sperm of monkey can fertilize sperm of ovum of monkey. Only sperm of a dog can fertilize ovum of dog. That is called a species specificity. Okay. Now outside this zona pellucida, you can observe these random cells here, which are sticking outside the secondary oocyte, and they are forming a sun-like appearance, the radiating sun-like appearance, and these cells are called as corona radiata. Okay, so these are the three layers present outside the cell. The first layer of egg cell is vitelline membrane, second layer of the egg cell is zona pellucida, and the third layer of the outside the egg cell, that is corona radiata. So when a sperm is entering or penetrating inside the egg, first the sperm will be penetrating the corona radiata layer, then it will be penetrating the zona pellucida layer, then it will penetrate vitelline membrane and get inside the cytoplasm, and then only fertilization will be completed. Okay. Now here I got a question for you all, statement based question. You can check the question. Statement 1. LH acts on the Sertoli cells and stimulates secretion of some factors which help in the process of spermiogenesis. Okay. Statement 2 is saying FSH acts at the Leydig cells and stimulates synthesis and secretion of androgens. Okay, now you have to answer, either the answer is 1, 2, 3 or 4. You can write down your answer in the comment section. Easy one, very easy question. You can write down the answer, and the timing I'm coming within 2 minutes. Just coming within 2 minutes. Okay, so all of you have answered as option 1. Both the statements are correct. How can you give wrong answer? Someone has said structural organization in animals. Yeah, structural organization in animals, it's done. You can check the previous lecture, or you can check the playlist of this channel. There you will be getting all the lectures, that is animal tissue, frog and cockroach. Okay. Now read the statements carefully. It's saying that luteinizing hormone acts on Sertoli cell. Is it so? No, my dear. Luteinizing hormone will be acting on Leydig cell to promote secretion of testosterone or androgens. So here it won't be LH, here it will be FSH. Sorry, here it will be FSH, and here instead of FSH it will be LH. So it will be vice versa. Sertoli cells are promoted or influenced by FSH hormone in male, and Leydig cells are influenced by luteinizing hormone. So that's why the answer will be both the statements are incorrect. You got the answer? Have you understood, Ishika, Niharika? Okay. Moving forward to the next question. Another question is here, the assertion and reason based question. And the assertion is saying the ovum retains most of the cytoplasmic content of the primary oocyte and is much larger than the spermatozoa. The reason is saying the ovum needs energy to actively seek out the spermatozoa for fertilization. Now the answer of this question will be, see, the ovum retains most of the cytoplasmic content of primary oocyte and is much larger than the spermatozoa. That is true, and the ovum needs energy to actively seek out the spermatozoa for fertilization. This is false, right? So actually it's sperm which is in need of energy for fertilization. So for this question the answer will be assertion is true but reason is false. Now next question, sorry, not next question, the next topic will be talking about, that is menstrual cycle. So let's get started. So when you talk about menstrual cycle, so the reproductive cycle in the female primates who are considered as female or primate, they are monkeys, apes like chimpanzee, gorillas, orangutans and human beings. We are also a primate animal, and female of these animals will be exhibiting menstrual cycle. But if you observe non-primates, if you are observing non-primate like a cat, deer, a cow, buffalo, they will not be exhibiting menstrual cycle. Rather, in their case, we can observe estrus cycle. That's a different phenomena and that's not given into our syllabus, right? So the first menstruation for human being begins at puberty and it's called as menarche. And the permanent end of menstruation and reproductive cycle and reproductive life, that is called menopause, and that ceases around 50 years. That ceases around means stopped around 50 years. Yes, exactly, Niharika. So in human females, menstruation is repeated at an average interval of about 28 to 29 days. That's equivalent to lunar month. So that's why the meaning of menstrual cycle, from where we got this term mens. Here men means not male. Here mens means is a Greek derived word that means month, right? So it's equivalent to a lunar month, and that's why it's repeated again and again. That's why we are calling it as menstrual cycle or monthly cycle. And this cycle of events starting from one menstruation till the next one is called the menstrual cycle. Okay, now we'll talk about the important phases of menstrual cycle. So what kind of changes you can observe during the time of menstrual cycle? So if you study menstrual cycle, you can observe some change which is happening inside the ovary, that is called as ovarian cycle. Simultaneously, you have to study the changes which is happening into the uterus, that is called as uterine cycle. So you have to study two distinct cycles or changes occurring inside female body. Next thing, and these are influenced by hormones. Firstly, they are influenced by hormones of anterior pituitary, and which are those hormones? They are LH and FSH. These two are hormones called gonadotropic hormones and secreted by anterior pituitary. Next, we need to take in consideration about the hormones which are released by ovary. So when you talk about ovarian hormones, and if we check inside ovary, it can produce very two crucial hormones required for menstrual cycle. One is estrogens, and another is progesterone. These two are the ovarian hormones. So we have to check these four phenomena: what kind of changes occurring inside ovary, what kind of changes occurring inside uterus, and obviously who are influencing them. Obviously the hormones of anterior pituitary, they are LH and FSH, and the LH and FSH, they are influencing the ovary to release ovarian hormones. They are estrogen and progesterone. Okay, so if we check the menstrual clock, if you check the menstrual clock, suppose the clock is started into this is the 0th day, means the beginning, and this is the middle of the menstrual month, that is 14th day, and here it will be 28th day. So total clock is of 20, the calendar is of 28 day. So up to fifth day, you can observe your uterus is bleeding. You can observe bleeding in uterus from first day to fifth day. You can observe uterine bleeding, slodding of the uterine endometrium layer. So that's why I am marking it red, right? First, second, third, fourth and fifth day, and this duration is called as menstruation phase. Okay. Now what after menstruation? After menstruation, what will happen? Generally, the uterine wall will start proliferation. Generally, the uterine wall will start thickening. So the second phase of this cycle is called as proliferative phase. Proliferative phase. Okay. Now at the age of 14th day, there will be ovulation, and after ovulation, what we can observe, the Graafian follicle is converted into yellow colored structure called corpus luteum, and it will die with forming a white colored corpus albicans. So from here to here, from 15th day to 28th day, this span of menstrual cycle, this is the third phase and called as luteal phase. So these are the three phases of menstrual cycle. First, from fifth day, where a female can experience menstrual bleeding, and that phase is called as menstruation phase or menstrual phase, the bleeding phase. Then from 6th to 14th day, from 6th to near about 13th or 14th day, where this uterus is continuing to thickening it up and preparing it for the next cycle, that is proliferative phase, the process of thickening of uterine endometrium, that is called proliferation. 14th day, the ovary will be kicking out the secondary oocyte, that is called ovulation, and from the 15th day gradually the Graafian follicle will get converted into yellow colored body called as corpus luteum. So this corpus luteum gonna sustain from the 15th day to towards 28th day, and that's why we call these as luteal phase. Now let's make a graph to understand the process in detail. Okay, so the x axis denotes the uh, days of calendar. So middle is 14th day, first, second, third, fourth, fifth, sixth, seventh, eighth, ninth, 10th, 11th, 12th, 13th. Then from here we got 15, 16, 17, 18, 19, 20, 21, 23, 24, 25, 26, 27, 28. Right, 26, 28. Okay, so this is the calendar from the first day to 28th day. Now we'll be dividing. First, we'll check the hormone titer of hormones produced by anterior pituitary. Then we'll check the titer of hormones secreted by ovary. Then we need to check the ovarian phenomena, and lastly we need to check the uterine phenomena or uterine cycle. Okay, first we will start with the uterine cycle. So the uterine cycle, first it will start with menstruation bleeding phase. Here the uterus, the thickness will be the least, and gradually from the sixth day it's getting or gaining more thickness, and again new cycle is started. So this is the thickness of uterus. This is the thickness of uterus to different phases of menstrual cycle, right? From first day to fifth day, from first day to fifth day, this is the menstrual phase. From fifth day, from fifth day to 13th day, this is the proliferative phase, and from 15th day to 28th day, this phase is called as luteal phase. So you can, if you observe, the lowest thickness of uterine wall that is observed during the time of bleeding phase or menstrual phase, the least thickness of uterus, and gradually during the time of proliferative phase, the uterine thickness has started uh, gaining it, and the highest thickness is observed during the middle of the luteal phase. So the thickest uterine endometrium is observed during the middle of luteal phase. Now what is the reason behind this? Okay, so this uterine phenomena, these are controlled by two ovarian hormones called estrogens and progesterone. So how estrogen and progesterone will work here? This is the graph of estrogen, the white graph that is of estrogen, and the yellow graph that is of progesterone. Okay, so basically the answer is lying here, why the uterus is gaining maximum thickness during the middle of luteal phase, is because during the time of luteal phase, the estrogen and progesterone, both of them are prevailing at their highest concentration. Estrogen is also high and progesterone is highest. So due to the simultaneous action of progesterone and estrogen, uterus will be gaining thickness, right? Okay. Now why after the bleeding, from the proliferative phase, estrogen, the uterine wall started gaining the thickness? Who is influencing this? See, this is influenced, the thickening of uterine wall is influenced by the estrogen hormone, right? So two questions are asked: which hormone is responsible for making the uterine endometrium thick? So who is building the uterine endometrium under the influence? The answer is estrogen hormones. And once uterine endometrium is thickened, is the duty of progesterone to maintain the thickness, right? One is building the uterus endometrium and another is maintaining the uterine endometrium. So that is the function of estrogen and progesterone. Yes, estrogen is building the thickness of uterine endometrium, and progesterone is maintaining the thickness of uterine endometrium. And as soon as the progesterone and estrogen is falling here, as soon as you can see progesterone and estrogen is falling, it will initiate the next bleeding phase or the next menstrual cycle. Now question is asked, why the graph of estrogen and progesterone is like this? Why there are two peaks of estrogen but there is only one peak of progesterone hormone. For that, we have to check inside the ovary now, because these two are the ovarian hormones. So first, inside the ovary you will be finding primary follicle, then you will be finding secondary follicle, then you will be finding tertiary follicle, then you will be finding Graafian follicle, and ultimately near to the day 14 the Graafian follicle will rupture. The Graafian follicle will rupture and secondary oocyte is released out. Okay. Now here I'll be explaining the first peak of estrogen. See, estrogen is produced by the follicular cells or the granulosa cells here into the ovarian follicles. Ovarian follicles containing some granulosa cells or follicular cells, these are responsible for secretion of estrogen. So as soon as this is the primary follicle, this is secondary follicle, this is tertiary follicle, this is GF, Graafian follicle. So as soon as the follicle is gaining the mass, more number of follicular cells, more production of estrogen. So the first peak of estrogen is simultaneous with formation of Graafian follicle, because Graafian follicle is the largest among follicle and it will be containing highest number of follicular cells or granulosa cells, and they will be producing high amount of estrogen. So it's ovarian follicle, they are primarily responsible for the first peak of estrogen hormone. Okay. Now gradually the Graafian follicle will ovulate, Graafian follicle will die. So gradually you can observe here the estrogen concentration is also falling. Again, what happens after that? Gradually corpus luteum is getting produced here. Once Graafian follicle is dying, so that will form corpus luteum. Now once this corpus luteum is produced, now that will be releasing progesterone, that will be releasing estrogen. So again once this corpus luteum is gaining the mass, it started secretion of progesterone, it started secretion of estrogen. So gradually again estrogen will take the second peak and progesterone will take the highest peak. So that is the reason behind this graphical representation of estrogen and progesterone. And gradually, why here there is a fall of estrogen and progesterone? The corpus luteum is dying here by forming corpus albicans, right? Okay. Now, which are the hormones which are influencing the differentiation of follicular phase? For that, we have to talk about the hormones which are coming from our anterior pituitary. Yes, and for our anterior pituitary, which hormones are coming? Is FSH and LH. It's FSH and LH. So first talk about FSH. First I will talk about FSH. So this blue line is of FSH. So gradually the FSH hormone is going towards the peak, and near to the 14th day FSH will show a rise or jump into the titer, and again gradually it will fall down. Okay. So with time, if you observe here, FSH is concentration of FSH is going high, and that will be stimulating the ovarian follicle to gain the maturity, right? So development of ovarian follicle, primary to secondary, secondary to tertiary, tertiary to Graafian follicle, is under control of which hormone? Under control of FSH hormone, which is coming from anterior pituitary, and that is the reason FSH is rising high, or ovarian follicle is gaining the mass. Ovarian follicle is gaining the mass, estrogen is also riding higher. That's why these two hormones are backing each other. FSH and estrogen are backing each other. High FSH, high estrogen. Low FSH, low estrogen. Okay. Now we need to talk about one more important hormone, that is luteinizing hormone. Okay. Now this is luteinizing hormone. So gradually luteinizing hormone will start to take the peak, and gradually it will fall down. Gradually it is falling down. So the yellow one, that is LH or luteinizing hormone. So luteinizing hormone, again it's going to its peak near to the 14th day. But if you check the peak of FSH, if you check the peak of FSH and peak of LH near to the 14th day, the peak of LH is quite higher than the peak of FSH, and that is called as LH surge. That is called as LH surge. Okay. Now this LH surge as well as high peak of FSH as well as this high peak of estrogen, all these trio, they are influencing the Graafian follicle to ovulate the secondary oocyte. So that's why this LH surge is very much important phenomena during the course of menstrual cycle, and when there will be LH surge, means 10-time jump, 10-time more concentration of luteinizing hormone into female blood just prior to this 14th day, just prior to ovulation. Okay. Now you can ask me a question, or I can ask you a question. Can you tell me that why there is LH surge? LH surge means the normal LH concentration, it's going 10-time higher, but for FSH jump, at FSH at the peak, the normal FSH concentration will go maximum 2 to 3 times, higher. If you compare to normal titer or normal concentration of FSH in female body. Now why there is no FSH surge but there is LH surge? What is the reason behind? What can be the probable reason? LH is rising high, but FSH cannot touch the peak of LH. Though in the beginning, if you observe, the concentration of FSH was higher than the LH, but at the middle of the cycle, you are observing LH is like a rocket and causing this LH surge. Can you answer me why there is LH surge, but why we cannot observe FSH surge? These two are gonadotropins, right? FSH is also gonadotropin. Basav Raju is saying FSH, both of them are gonadotropin. FSH is also gonadotropin, LH is also gonadotropin. But why LH is out rocketing but FSH is unable to catch the peak? What can be the reason? Is there anybody who can answer? Want to answer this question? Okay, let me tell you. So once Graafian follicle is forming, the ovulating the secondary oocyte, sooner it will get converted into what? Sooner it will get converted into corpus luteum. Okay. Now you know that this follicular stages can produce one more hormone that is called as inhibin. This follicular stages can produce, these stages can produce estrogen, these stages can produce estrogen as well as progesterone. But this follicular stages can, yes, Ishika, they can produce one more hormone called inhibin. Now it's inhibin, it's trying to low down, it's inhibin trying to drag, trying to grab the concentration of the FSH towards lower side. So that is the reason there is surge of LH, but there is no such surge of FSH can be seen. But if you eliminate inhibin from your blood, if you are a female, then you can observe FSH surge as well. Okay, so we are done with menstrual cycle. Now let's check what NCERT is telling about menstruation. So menstruation only occurs if the released ovum is not fertilized, right? So lack of menstruation may be indicative of pregnancy. If you are missing menstruation for 1 month, so ultimately everybody is thinking that you are pregnant. However, it may also be caused due to some other underlying causes like stress, poor health and unhygienic condition. They can cause an amenorrhea or irregular menstruation. Okay, during pregnancy all events of the menstrual cycle stops and there is no menstruation or menstrual bleeding will be occurring during the time of pregnancy. So in the absence of fertilization, the corpus luteum degenerates. As I say, in normal months corpus luteum can only sustain for 10 to 12 days, but during the time of pregnancy fertilization takes place, corpus luteum can sustain for 3 to 6 months, right? Okay, so this causes disintegration of endometrial lining. If corpus luteum dies, then that will cause deterioration of progesterone level as well as estrogen level, and that will cause the menstrual bleeding or menstruation and marking a new cycle. The cyclic menstruation is an indicator of normal reproductive phase and extends between menarche to menopause. Mean the reproductive phase of a female, the beginning should be from menarche and the ending should be from menopause. Okay, maintenance of hygiene and sanitation during the menstruation is very much important. Use of sanitary napkins or clean homemade pads. Change sanitary napkins or homemade pads after every 4 to 5 hours. Why it's so? Because within 4 to 5 hours there is a tendency these sanitary pads or napkin will start generating, will start infecting by some microbes. Okay, that's why within every 4 hours you have to change sanitary pads or napkins as per the requirement. So disposal of those used sanitary napkins, properly wrapping it with is properly used paper. So disposal is another sustainable practice. If you are throwing the used sanitary napkins anywhere or sanitary pads, so they are containing some gel kind of structure. Okay, if you cut the sanitary pad, you can observe some bluish colored gel kind of structures are present inside that to absorb the excessive fluid, right? But if you are throwing directly on the mud, it gonna create soil pollution, and sometimes it's carcinogenic, cancer causing also. That's why proper disposal of used napkins or used sanitary pads is a sustainable practice for us. Okay. Now the next topic we are jumping in, that is fertilization and implantation. So fertilization can only occur if the ovum and the sperms are transported simultaneously to the ampullary region. So the region where fertilization is taking place, that is ampullary region or ampulla of fallopian tube, and a terms and condition is given here. The sperm and the ovum or the secondary oocyte should present at a similar point of time, right? It's very ironical thing that once a male is inseminating the semen into the female reproductive system. Gradually the sperm will be swimming towards the fallopian tube and will be going into the ampullary region of fallopian tube and resting and waiting for the ovum for 2 days, nearly 48 hours. That's why we say that sperms can survive into the female reproductive tract for 2 days or 48 hours. But this ovum after ovulation will enter into the fallopian tube, and the ovum or the secondary oocyte is having a typical girl nature. See, a boy, always you can observe a boy is waiting for many days for a girl to respond, but girls are always with attitude, right? Yes, Ishika. And the girls are always in attitude. Similar thing is observed with the behavior of the secondary oocyte. Upon ovulation, it will enter into the fallopian tube and it will wait only for 12 to 24 hours. Then it will degenerate. Okay, that's why the timing is very much important, and this is the reason why not all copulations lead to fertilization and pregnancy. Okay. So, what are the different phases of fertilization? You all know that fertilization means the fusion of sperm and ovum. But we have to talk about the different phases while gradually the sperms are approaching towards the ovum. See, it's a kind of inspirational journey of sperm. When a man is inseminating semen into the vagina of a female, then see what the vaginal environment, the vaginal content is acidic in nature, but acid is toxic for sperm. So then and then 90% of sperm gonna die into the vaginal cavity. Okay. Now somehow 10% of the sperm will be surviving from vagina, and through the cervix they will enter into the uterus. Few sperm will go or thrive toward the left fallopian tube. Few sperms will be thriving toward the right fallopian tube. Now chance is there that only one fallopian tube, the ampullary region, will be containing the secondary oocyte. Now can you tell me that out of those 200-300 millions of sperm, ultimately how many sperms can reach near to that secondary oocyte staying or lying into the ampullary region? Niharika, if you are getting the option ampullary region and ampullary isthmic region, you have to choose for ampullary region because that's a direct line of NCERT. Here we cannot do anything, but what you said, that is correct. The exact place for fertilization frequently we can observe, it's actually ampullary isthmic junction where the fertilization takes place. But as NCERT is saying, it's ampullary region, we have to mark ampullary region only. So I was asking a question, can you answer that? Out of the 200-300 millions of sperm, no, only one can fertilize the secondary oocyte. But how many of the sperms can reach or can visualize the secondary oocyte inside the ampullary region? They are few hundreds. Okay. So out of those 300 million, 99.99% they will die. 0.1% will be reaching near to the secondary oocyte, and out of the 0.1%, only one, the strongest or the chosen sperm, can fertilize with the ovum. And this is a new discovery. We say that the fastest and the strongest sperm can fertilize the ovum, but it's not the exact phenomena. It's the ovum who is choosing the sperm, right, with the help of some chemical coordination, chemical interaction. Okay. So if we think about different phases of fertilization, so first what will happen? The sperm will go near to the secondary oocyte and it will bind with the zona pellucida. So first the sperm binds to zona pellucida of secondary oocyte. Now this binding of sperm with the secondary zona pellucida of secondary oocyte will trigger a change of polarity inside the ovum. In normal case the ovum is negatively charged. But once the sperm is binding with the zona pellucida, the ovum will become positively charged. That is called change in polarity. Okay, so there will be change in polarity of secondary oocyte, and this is a very miraculous event. This polarity change will repel all other sperms. This polarity change will repel all other sperm except the lucky one, because already it is attached to the zona pellucida, because the normal polarity of sperms are positive. So this positive secondary oocyte will repel all other sperm and except this lucky one which is already attached and performing synagamy with this secondary oocyte. So this change of polarity is in order to block the attachment of some unwanted sperm which are swimming nearby region. So this phenomena is called as fast block. It's blocking very quickly, it's blocking the unwanted sperms. Now third phase will be there. Now gradually the acrosome will release the enzyme, so that is called acrosomal reaction, and gradually the sperm will penetrate into the ovum. Sperm is penetrating into the, it's not ovum, it's secondary oocyte. Okay, and release the haploid nucleus. Sperm will enter into the cytoplasm and will release the haploid nucleus, and both the nucleus are dancing with each other to bind to form a diploid cell. Right? Okay. So this phenomena is called as syngamy. Now next what will happen? Next, this cortical granules, you know about cortical granules present into the cytoplasm, will be released into the perivitelline space, and one more layer of wall will be formed in between zona pellucida and vitelline membrane. The fifth stage is called as cortical reaction. And in this cortical reaction, a special wall or membrane is formed outside the vitelline membrane called as fertilization membrane, and that is further blocking all other disturbance from the outside world, because already it has taken the lucky sperm inside. And one more important thing, for mammalian fertilization, complete sperm will be entering into the ovum. Generally we see that in different books it's written that sperm will only be penetrating its head region, right, inside the cytoplasm. That's about non-mammalian sperm. But for mammalian sperm or human sperm, during the time of fertilization, head, neck and tail, 100% of sperm gonna penetrate or get inside the ovum. Because see, ovum is a large cell. The size of ovum 100 micron and the size of sperm 5 micron. So easily the human sperm can completely enter by penetrating all this layer and perform the process of fertilization. Okay. Now during the time of fertilization, what are the different phenomena will be performed? First, there will be a combination of the nuclear material, that is called as karyogamy. Karyogamy, nucleus of sperm and nucleus of pronucleus of sperm and ovum gonna merge with each other. That is called karyogamy. Second, there will be plasmogamy. Plasmogamy, the cytoplasm of sperm and the cytoplasm of ovum will be mixed with each other. During the time of plasmogamy, the sperm will be donating centriole to the ovum, and in the meantime the secondary oocyte is also will be converted into ovum or oocyte. So that's why we say that centriole is paternal in origin, means every cell is having centrioles and those centrioles are coming from our father. Every cell is having mitochondria and the mitochondria are coming from your mother. So centrioles are result of paternal inheritance, mitochondria or mitochondrion, they are result of maternal inheritance. So it's sperm who donates the centriole and the haploid nucleus to the ovum. Rest everything present inside the ovum only. So karyogamy, mixing of the nucleus, plasmogamy, mixing of the cytoplasm, and ultimately chromosomal exchange in between the nucleus of ovum and sperm, that is called amphimixis. Amphimixis is the process of chromosomal exchange of the genetic material of ovum and sperm. So that was all about fertilization. Now we'll talk about implantation. Now once the zygote is formed, which is diploid in nature, now that will start a special kind of mitotic cell division, but that is quite different from mitosis. Okay, and the cell division is called as cleavage. The cell division is called as cleavage. Okay, now what is difference between mitosis and cleavage? In mitosis, if this is the mother cell, two new daughter cells are formed and they are of similar size in compared to the mother cell. But in cleavage, what happens? In cleavage, if this is the mother cell, when it will be dividing to form two daughter cell, the cell will be reduced in their size. Though all are diploid in nature, if this is the scenario, this is called as mitosis. From one diploid cell to two diploid cell of similar size, this is simple mitosis. But from one diploid cell to two diploid cells are forming, but new cells, means daughter cells, are reduced in size in compared to the mother cell, then that special kind of mitosis is called as cleavage. Remember, so the zygotic division is not normal mitosis, it's called cleavage division. So first cleavage will be there will be formation of two cells. Then another cleavage will be there, second cleavage will be there will be formation of 4 cells. Another cleavage will be there will be formation of 8 cells. Another cleavage will be there will be formation of 16 cells. Another cleavage will be there will be formation of 32 cells, and so on, right? So this this primitive cells of our embryo, these are called as blastomeres. These are called as blastomeres. After the zygote, 2 cell stage, 4 cell stage, 8 cell stage, 16 cell stage. So each cells are called as blastomeres. They are kind of totipotent cells or pluripotent cells. Now can you tell me what do you mean by totipotent or pluripotent cells? I am saying these blastomeres are either totipotent or pluripotent in nature. Now, can you tell me in the comment section what do you mean by totipotency or pluripotency? That means they have the capability to produce complete body. Okay. Now, from these developmental phases, particularly 8 to 16 cell stage of embryo, we call them as morula. We call them morula. So this is the morula stage. Why it's called as morula? Because the size, the shape, sorry, the structure of this 8 to 16 cell stage of embryo, it looks like a bunch of mulberry fruit. That's why we call it as morula. And from 32 cell stage onward, from 32 cell stage onward, we call these as blastocyst. We call this as blastocyst. Okay, and this is the state which is gonna be implanted, into uterine endometrium. Uterine endometrium. So which stage of embryo is getting implanted into uterine endometrium is the blastocyst stage. What is the chronological order of zygotic development? Zygote will divide into 2 cell stage, then 4 cell stage, then 8 cell stage, then 16 cell stage. Now this 8 and 16 cell stages are called as morula stage. While this morula will be dividing to form more cells, we will be calling these as blastocyst. Now why this is named as blastocyst? Why it's so got so much unique name? It looks like a football. It looks like a football with some peripheral cells and some inner cells or inner cell mass. So that's why this hollow football kind of structure with the peripheral cells are called as trophoblast cells, and the inner cells are called as inner cell mass, ICM, inner cell mass, along with the cavity inside that is called as blastocele. It's a football kind of structure which is gonna be implanted soon, which is covered by zona pellucida before implantation. Still the zona pellucida will be prevailing outside the blastocyst just prior to implantation, right? So this hollow ball kind of embryonic structure, we call it as blastula or blastocyst. Why it's called as blastocyst? Because of the presence of this tumor kind of structure or cyst kind of structure called as inner cell mass into the hollow cavity. Yes, Ishika. Totipotent cell have ability to produce all type of cell. Exactly. Okay. So now what will happen gradually? See, what I said, the blastocyst is always covered by zona pellucida. If this is blastocyst, stage, gradually that will come out of the zona pellucida layer. If this is the blastocyst stage, gradually that will come out of zona pellucida layer, and that will form some finger-like projection into the trophoectoderm or this trophoblast or the outer cell part. These are called as chorionic villi. So, with the help of this villi or this finger-like structure, with the help of this villi or this finger-like structure, this blastocyst will get implanted. This blastocyst will get implanted. Suppose this is the uterine endometrium, will get implanted into the uterine endometrial layer. Okay. So that's how implantation is taking place, and the beginning of the class, someone was asking, I think Prajwal was asking that, sir, how twins are formed, and someone else was asking, sir, what do you mean by anterior placenta and posterior placenta? Okay, now I can give those answers. See, first I will answer how twins are formed. How twins are formed. See, twins are of two types: homozygotic twin and heterozygotic twin, fraternal twin and maternal twin. So, first, twins which are homozygotic means they are identical to each other. Either they can be both are male or both are female. Identical twins cannot be like this, one is male, one is female. For identical or homozygotic twin, they look quite identical. They either they are both of them are males or both of them are females. So how it's formed? See, when this blastocyst stage is coming out of zona pellucida layer and preparing for implantation, it's cutting that zona pellucida layer and it's coming out by forming a 8-shaped structure, right? And that is called hatching process. When this blastocyst is coming out of zona pellucida covering, we call this process as hatching. During the time of this hatching process, if it's too much twisted, then what it will do, it will be splitted into two blastocysts. It will be splitted into two mass of blastocyst. Now from this it will make a baby, and from this it will make another baby. So these two babies are exactly identical to each other, and as because they are originated from the same zygote, same blastocyst, we call them homozygotic twin. Now what is the funda of heterozygotic twin? Heterozygotic twin, that's little bit less complicated. We know that for normal condition, one ovum, one secondary oocyte is ovulated by one healthy female, right? But possibilities are there that in a single month a female can ovulate more than one secondary oocyte. So suppose if what will happen if a female for a certain month is ovulating 2 secondary oocyte or ovulating 3 secondary oocyte. So in that case, 2 different sperms or 3 different sperms, or maybe from both the side, both the ovaries have released 1 secondary oocyte. So there will be 2 secondary oocyte and 2 different sperms are gonna fertilize them, and there will be formation of 2 zygotes and 2 babies. So this is the condition of heterozygotic twin or non-identical twin or fraternal twin. It's mentioned in the exercise part of this chapter, and the last year we got the question as well in NEET. What is fraternal twin? Okay, they are heterozygotic twin. Why we are calling them fraternal twin? When two different sperms, when two different sperms will fertilize two different ovum inside the female body, and then they will give rise to different zygote and two different embryo and two different babies. It may be possible that one is male, one is female, or both of them are male or both of them are female. Is an example of fraternal twin. Example of what? Fraternal twins. Now, if you are getting a question like this in your class, uh, uh, you have two friends, they are one brother and one sister, right? The name of the brother is Sonu, name of the sister is something, suppose it's Ishika. Sonu and Ishika, both of them are brother and sister, and they are twins. Now you have to tell me either they are heterozygotic twin or they are homozygotic twin. So they will be heterozygotic twin or fraternal twin. When the twins are one male, one female, then blindly you can answer they are fraternal twin or heterozygotic twin. But when both of them are male or both of them are female, then possibilities are there that they are maybe identical or homozygotic twin. Have you got the concept? Have you got the concept of twins? Or you wanted to ask anything regarding the concept of twin formation? Is there anyone who want to ask anything from the concept of twin formation? Heterozygotic twins, yeah. So heterozygotic twins are called as fraternal twins. See, there is a copulation between one male and one female. That male partner will be releasing millions of sperms, and luckily in that particular month that female partner ovulated two secondary oocytes instead of one secondary oocyte. So though that's a rare case, but that can happen, right? Okay, that is called superovulation. So in that case, what will happen? Inside the reproductive system of that female, you will be finding two secondary oocyte. Inside the reproductive system of that female, you will be finding two secondary oocyte. Now, as because multiple sperms are there into the semen ejaculum, so two different sperms are gonna fertilize those two secondary oocytes, and from each fertilization there will be formation of one zygote. From this fertilization there will be formation of another zygote, and this will form one baby. This will form one baby, and this will form another baby, right? So they are called as heterozygotic twins or fraternal twins. Okay. Now I am coming to the placentation part. Now, how these embryos are attached to the uterine endometrium? These embryos are attached with this uterine endometrium with the feto-maternal tissue, called as placenta. Called as placenta. So what kind of tissue is this? Placenta is feto-maternal tissue. What do you mean by feto-maternal tissue? Means it's a kind of bridge in between that embryo and the mother, and the placenta is made, the tissue is shared by the embryo, shared by the fetus as well as the mother. That's why half of the placenta, the tissue will be made by mother, and half of the placenta, the tissue will be given by the fetus. So that's why it's called as feto-maternal tissue. Basically, it's acting as a filter in between the fetus and the mother. It's filtering good materials from mother to fetus body, and inhibiting or preventing the entrance of harmful materials from mother blood to fetus. So that is the basic function of placenta. And when you talk about placentation, means which is the good place of uterus where this placenta can be formed, or placenta the fetus or the embryo can be embedded. So if we talk about the structure of uterus, so the outer part, the upper part is called the uterine fundus. So it's like this, right? So this uterus fundus is considered as the best part for placentation or the implantation, or the good places for implantation can be the above uh, midline of the uterus. Means if implantation is occurring into this part, that's also good. If implantation is occurring into this part, that's also good thing. If implantation is occurring into this part, that's also good thing. If implantation is taking here inside the fallopian tube, that's bad thing. If implantation is taking near to the cervix, that's also a bad thing. That is called as ectopic pregnancy. That is called as ectopic pregnancy. This is example of ectopic pregnancy. This is example of ectopic pregnancy. This is perfect form of pregnancy. This is perfect form of pregnancy. Sir, is any disease can also transfer from placenta from mother to fetus? Yes, there are many diseases are there that can be transferred from mother to fetus. Like if we talk about HIV/AIDS, they can be transferred from mother to fetus. If you talk about hepatitis B, remember these two diseases, HIV/AIDS and hepatitis B, these two diseases can be transferred from placenta to fetus. Now, if you talk about the erythroblastosis fetalis, that is another condition, right? The Rh incompatibility in between mother and the second child onwards, that mother's immune system can attack the Rh antibodies, can attack the RBCs of the fetus, and that may cause damage of liver, anemia, and the condition is called as erythroblastosis fetalis. So though placenta is trying to prevent all kind of infection or harmful things to reach or to contaminate the baby, but sometime few pathogens or few immunological components can cross the placental barrier. Suppose if mother is consuming cigarettes or tobacco, if mother is consuming, if mother is taking narcotics, drugs, if mother is consuming alcohol, then those toxic elements can cross the placental barrier and cause disease in fetus as well. Okay. And someone was asking about anterior placenta and posterior placenta. Though it's not given in the syllabus, but if you check the pose of the body, the back side is your posterior side, the front side is your anterior side, right? Now, if this is the womb and if placenta is formed towards the dorsal side, then that is called as posterior placenta. And if placenta is formed near to the belly side, that's called as anterior placenta, though it's out of our syllabus. Someone was asking that time, that's why I'm giving the answer. Okay. Now let's check what NCERT is saying. So immediately after implantation, the inner cell mass of the blastocyst will differentiate into an outer layer called ectoderm and the inner layer called as endoderm. So during the time of cellular division, first ectoderm and endoderm is formed. We know that we are triploblastic animal. We have ectoderm, mesoderm and endoderm. But first, during the time of embryonic development, first ectoderm and endoderm is formed, and after that gradually the mesoderm soon appears in between the ectoderm and endoderm. This is very important statement you need to remember. They will not be arriving or appearing simultaneously. Mesoderm appears after the formation of ectoderm and endoderm. Now these three layers give rise to all kind of tissue or organs into our adults. Like if you talk about ectoderm, it can give rise epithelial tissue, it can give rise nervous tissue. If you talk about mesoderm, it can give rise connective tissue, muscles will be formed by mesoderm, right. If you talk about endoderm, it can give rise your blood vessels or few kinds of uh, mm, epithelial layering. Okay. So it needs to be mentioned here that the inner cell mass contains certain cells called stem cell. Very important statement to remember. So stem cell, we have seen that this blastocyst, it had one outer cell mass, outer cell called trophoblast or trophoectoderm, and inner cell mass. So basically these outer cells, they are responsible for formation of placenta and implantation, and the inner cell mass, they are responsible for formation of the body of that embryo or the baby. And that's why we are calling them, we are naming them as stem cell, because these are the cells which can give rise different kind of tissues and organs and systems. So which have the potency to give rise to all the tissue and organs. Already we were discussing about totipotency or pluripotency. So we can say that these cells of inner cell mass, they are either kind of totipotent cell or they are kind of pluripotent stem cell. Okay. So this is the diagram of placenta, and embedded placenta is given in NCERT. Now here we are going to talk about some placental hormones. Placental hormones. What are placental hormones? Hormones secreted by placenta. They are HCG, human chorionic gonadotropin hormone, and this HCG is a very crucial hormone after the placentation or after the gestation, right, as because it's HCG which will be signaling, giving signal to the corpus luteum of your ovary to sustain for 6 months. Otherwise the corpus luteum will die within 10 days or 11 days, you know, right. But during the time of pregnancy, the same corpus luteum will survive for 6 months at least, and that is boosted, that survivability is promoted by HCG, human chorionic gonadotropin hormone, produced by placenta. Another important hormone released by placenta, this is called HPL. Okay, human placental lactogen, that will be helping in maintaining the metabolic reaction of female, the mother, or that will also be influencing the mammary gland for its proliferation. Then the hormones secreted by placenta, that's relaxin, that's estrogen, progesterone, and corticotropin releasing hormone, CRH. So these are the important hormones produced by placenta. Again I am repeating, HCG for human chorionic gonadotropin, HPL for human placental lactogen, relaxin which will be required for during the time of baby delivery, estrogen and progesterone. Yes, high concentration of estrogen and progesterone secreted into blood through the placenta, and CRH, another hormone required during the time of parturition or baby delivery, that is corticotropin releasing hormone. Okay, I got a question, sir. What are the safe dates to have intercourse to avoid pregnancy? And is it possible to get pregnant if the intercourse happen while menstrual cycle? See, chance is there. See, chance is there, but you cannot say that if you are performing coitus during the time of bleeding period or menstrual period, uh, there won't be any pregnancy. But yes, safe period is there, that is called fertile window. Okay, let me explain you that particular thing. So we know that what is the duration of menstrual cycle? That is 21 day, from first day to 28th day. First day to 28th day. What is the middle of the cycle? That is the 14th day. So if your menstruation duration is 28 day, you have to minus 14 and you will be getting the 14th day. That is the day of your ovulation. That is the day of your ovulation, right? Now what you have to calculate here, you have to do plus 3 and minus 3 with that 14. So if you minus 3 for 14, you will be getting the 10th day. If you add 3 with the 14, you will be getting the 14, 16, 17, 18. Let me take 17th day. So this 10th day to 17th day, if someone of the menstrual month, if within this span or within this window someone is performing coitus or sexual intercourse, then there is highest chance of getting pregnancy, and that is called as fertile period or fertile window. 3 days before to the ovulation day and three days after to the ovulation day. And here let me tell you one more interesting fact about the menstrual cycle. Suppose for one female, the menstrual month is of 28 days. The gap between one menstruation and another menstruation is 28 days. So menstrual month will be 28 days. For another female, suppose the menstrual month is for 34 days. So what will happen? What will be the fertile window in case of that particular lady? So what you have to do, you have to again minus 14 from 34. So you will be getting what? 34 minus 14, that is 20th day is the date of ovulation for her. Okay, the menstrual month duration is not equal for everyone. It will be differing from female to female. For a particular female, the menstrual month duration can be of 25 days. For another female, the duration of menstrual month can be of 30 days. So 28 is the average date. So that one particular female can calculate accordingly, right? So if for her a menstrual month is of 34 days, it's natural for her, it's regular for her, then after minusing 14, you are getting 20th day is the day of ovulation. So again you have to do plus 3 minus 3. So for her, the fertile window will be in between somehow from 16th day to 23rd day. So that's how it's calculated. Safe dates are calculated. You have to avoid this fertile window, and in the next chapter you are going to study that. That is the calendar method or the periodic abstinence method, right? So I was talking about these placental hormones. You need to remember these names of placental hormones. Okay, now we'll talk about one more important thing. It's many time it was asked that what are 3 exclusive pregnancy hormone? Means hormones which are produced only during the time of pregnancy. Firstly, HCG, only produced during the time of pregnancy. Secondly, HPL, only produced during the time of pregnancy. And thirdly, the relaxin. These 3 are the exclusive hormones which are produced only during the time of pregnancy. Yes, Niharika, I am coming to that part. Thyroxine and prolactin, they are not released by placenta. What I was talking, I was just discussing about the placental hormones. So these are the hormones which will be secreted by placenta. Next I told that these 3 are the hormones which are secreted exclusively during the time of pregnancy period. Now next slide I am going to tell you that all the hormones you required in high concentration in female blood during the time of pregnancy. Okay, so which are the hormones? Is progesterone, is estrogen, actually it's estrogens. Estrogens are actually group of hormones. Then we need high titer of cortisol. We need high titer of thyroxine or thyroid hormones, right? We need high titer of prolactin hormones. So these are the essential hormones which is maintained into high concentration or high titer during the time of pregnancy or gestation period. During the time of gestation period. Okay, now you got my point, Niharika. So if you are asked about placental hormones, you have to talk about ACG, HPL, relaxin, then estrogen, progesterone and CRH. Okay, if you are asked about name the 3 hormones which are exclusive during the time of pregnancy, HCG, HPL and relaxin. Name the hormones overall in female body which you require to maintain the high concentration in female blood. There this sort of hormones, obviously thyroxine and prolactin and cortisol, these 3 hormones are not produced by placenta. Yes, placenta can produce progesterone and estrogens. Okay, now the next topic we'll be covering, that is what are the major features of embryonic development at various months of pregnancy. Okay, so the major phenomena, the features we can observe during the time of morphogenesis, means gradually the fetus, the embryo is developing different internal organs or external structure. So let's talk about that. So NCERT is saying that the total 9-month duration is divided into 3 subsequent parts and of 3 months, 3 months and 3 months, and we will be calling them as trimester. First 3 months we will be calling them first trimester, then 4th, 5th and 6th months second trimester, 7th, 8th and 9th months is called as third trimester. Okay, so NCERT is telling about only first month, second month, third month, 5th month, 6th month and 9th month. What are the important morphogenetic phenomena happening inside the fetus? Okay, so see, for the first month what will happen? You have only one heart. Remember, focus it in a proper manner. You have only one heart into your body, so it's the one heart, and the first month there will be formation of the heart, and the first sound of the heart, the lub-dub sound with the help of a stetho can be heard. So that's very quite easier to be remembered. One heart, and when the heart is formed, first month. Okay, now let's come to the second month. This 2 means second month. See, it's written there is formation of limbs and digits. There is formation of limbs and digits. Okay, so second month, how many hand do you have? How many hands do you have? You have two hands. So the baby is saying already has launched into the mother body. So baby is showing victory. Baby is showing victory. In the second month, there will be formation of digits, and digit means fingers and toes, and limbs means hands and legs. Okay. So third month, there will be formation of genital organs and important systems inside the body. So how to remember that? Third month, there will be formation of genital organs and important systems. So genital organ means sex. So in human being, how many sexes are there? Male, female and transgender. 3 sexes are there, right? So in the third month, there will be formation of, you can remember in this way, genital organs and other important systems will be formed. Yes. Now let's come to the fifth month. Fourth month is not mentioned in NCERT. So fifth month, what will happen? The first movement of fetus and formation of hair on head will be there. See, the first movement of fetus, how fetus will move? Remember, the fetus will move like this. Fetus will move like this. Fetus will move like this. Okay. 5 for F, first for F. So on fifth month, first movement of fetus will be there, and fetus is moving like this. So basically that means it's showing hair is formed on head. Okay. Next, let's come to the sixth month. What will happen? Eyelids are separated and eyelash is formed. Eyelids are separated and eyelash is formed. See, sixth month means second trimester. Sixth month means end of second trimester. We have two eyes. So in this manner, you can remember in the end of second trimester or sixth month, the fetus can blink the eyes, right? So its eyelids are separated and eyelash, and in the verge of 9 months is the end of the third trimester, the fetus is ready for parturition. So we are done with the major embryonic developmental phases. Now we will be talking about the method of parturition. So the average duration of human pregnancy is about 9 months, but somehow in real world the parturition can take place in between 7 months to 9 months, which is called the gestation period. Various contraction of the uterus, vigorous contraction means very fast and urgent contraction of uterus at the end of pregnancy causes expulsion or delivery of the fetus, and this process of delivery of the fetus is called childbirth or parturition. But if you study NCERT properly, it's asked there, can you tell us that what is the gestation period for dogs, cats and elephants? Can you write down in the comment section what is the gestation period for dog, cat and elephant? Okay, so till how many month? Till how many month one can perform abortion? It's recommended. It's within 2 months. Okay, or within first trimester. If you are performing abortion, that comes under the MTP Act. Okay, so parturition induces by a complex neuroendocrine phenomena. So why it's said as neuroendocrine mechanism parturition? Because during the time of parturition, you need some reflexes called as fetal ejection reflex, which will be produced by our nervous system. So that's a neural mechanism, and that will be associated by some hormones called oxytocin relaxin and prostaglandin. These three are the major hormones which are required during the time of parturition. Remember, they are oxytocin, oxytocin, prostaglandin and relaxin, which are must during the time of childbirth or parturition. So that's why it's called as neuro as well as endocrine mechanism. Okay. For Ishika is saying for dog it's 58 days, for cats 58 days similar. Okay. For cats and dogs it's near about 2 months. Okay. For elephant it's near about 2 years. Yeah, 22 months. Okay. Niharika is saying also giving the right answer, 62 to 64 days. That's great. Yes, exactly. For dog and cat, it's nearby 60 days. Okay, 2 or 3 days more or less, plus minus 2 months. And for elephant, it's near about 2 years, right? Plus minus 3 months. Okay. So how parturition is happening? So the initiation of parturition will be initiated by developed a matured placenta and the fetus. When the fetus and the placenta is fully matured, that will create one neurological urge that is called fetal ejection reflex. That is called as fetal ejection reflex. Mature developed placenta and fetus will produce a fetal ejection reflex. It's a kind of neurological urge of your body to deliver the baby. It is called fetal ejection reflex, and that will create mild contraction. It will create mild contraction of uterus. Okay. And this mild contraction of uterus will give a positive feedback to maternal hypothalamus and maternal posterior pituitary. That will give a positive feedback to maternal hypothalamus and posterior pituitary. And you know that hypothalamus can produce oxytocin, and through posterior pituitary oxytocin is released. So there will be release of oxytocin, and you know that this oxytocin can bind with the smooth muscle receptor of uterine myometrium, and that will cause increased or faster or stronger contraction of uterus, a contraction of uterine wall. Now, what will happen after that? Uterine wall is contracting vigorously, strongly. Now, that will creating a push outside towards the outside the body. Okay, and that is called as expulsion phase. That is called as expulsion phase. So there will be expulsion of fetus, means the fetus will be literally kicked out from the uterine cavity to outside the body. That is called fetus expulsion of fetus. Okay, now while this fetus is coming out of the body, you need help of some hormones. Obviously, one hormone we have spoken about, that is oxytocin, that is responsible for uterine contraction. But while the fetus is coming out through the birth canal, through cervix, through vagina, you need to dilate, you need to widen the opening. You are welcoming the baby into the real world. For that, you need another hormone called as relaxin and prostaglandin. So these hormones are required for parturition purpose, and once the fetus is expelled out, still the placenta is tucked inside the mother's womb. So the last phase will be like this: expulsion of placenta. Now the baby is coming out with the umbilical cord. The doctor will cut that umbilical cord and will keep the baby into a safer condition. Okay. Now one more thing is important just after the delivery, and what is that? That is, once the baby is freshly delivered, the baby will not show any kind of reflex or any kind of response, because its respiratory system is not working. So suddenly you will observe that doctors are patting on the chest or patting on the back, so that the fluid, the amniotic fluid which is accumulated into the respiratory tract of the baby is gradually coming out, and the baby will start taking breath, and whenever the baby is breathing or exhaling for the first time, that baby will start crying, and that is the signal that baby is alive. Okay. Next, now it's time for the baby care, nurturing the baby with the breast milk of mother. The mammary glands of the female undergo differentiation during pregnancy and starts producing milk towards the end of pregnancy. And you know that it's the prolactin hormone which will be which is responsible for influencing the mammary alveoli to produce the milk by the process called the lactation, and this helps the mother in feeding the newborn. And you know that which hormone is responsible for ejection of milk? This is oxytocin. As basically when a baby is breastfeeding his or her mother, then there will be a connection, there will be an emotional bonding, formation of emotional bond in between the child, in between the infant and the mother, and that will start release of more and more oxytocin, and that will help mother to release more milk. Okay. So the milk produced during the initial few days, and that's another important line of initial 4 to 5 days of lactation, the mother will be producing a yellowish milk which is very condensed and called as colostrum, which contains several antibodies, like which contains IgA, right, absolutely essential to the develop resistance for the newborn baby. And what is the difference between colostrum and normal milk? Colostrum is rich in protein and there is less fat. For normal milk, there will be less protein and more fat will be there. Okay, so we are done with this chapter. Now just try if you can answer this question. After the formation of the zygote, the cleavage it cleaves into 2, 4, 8 or 16 daughter cells, and these 2, 4, 8 or 16 cell stage are called as blastocyst, morula, trophoblast or blastomeres. Can you answer this? Okay, you try to give the answer and I will be coming back within 10 minutes. Now here it's a break. And after that we will be starting the new chapter, that is reproductive health. Yes. So, what do you want to know about Arjuna NEET English, Kunal? Can you please write down in detail? ऐसी ऐसी नहीं चल रही, वो कुछ नहीं होगा। बंद है, बंद है। Okay, so I'm back with another chapter from the unit reproduction, and the chapter is reproductive health. So let's get started with this chapter. So we know that when you talk about health and disease and all these things, so it's regulated by WHO, World Health Organization. Yes. So according to WHO, if you are physically fit, you already know that, then who will not be saying that you are healthy or reproductively healthy. But if you are maintaining all these criteria, you are physically healthy, emotionally healthy, behaviorally healthy and socially healthy, then according to who you are qualifying, and we can say that you are reproductively healthy person. So what do we understand by this term? The term simply refers to healthy reproductive organs with normal function. So according to World Health Organization, reproductive health means a total well-being in all these aspects. And which are these aspects? Physical, emotional, behavioral and social reproductive health. Okay. Now, when you talk about this chapter, and if we take a look through the different countries of the world, so we will be finding that India was amongst the first countries in the world to initiate action plans and programs at a national level to attain total reproductive health as a social goal. And you know that under that campaign, we know about family planning program which was initiated by Indian government in 1951. You need to remember this year of family planning, and later on there was a drastic modification, and through different phases of modification now we don't know about family planning, that is changed into another name, RCH, Reproductive and Child Health Care Program. So what is the abbreviation of RCH? Reproductive and Child Healthcare Program. What is the motto of RCH? Is to create awareness through different audio visual medias, and providing facilities and support. Not only you will be providing awareness, not only you will be explaining them about good things or bad things, but you have to provide facilities like hospitals, doctors, medications, and ultimately motto is to build reproductively healthy society. So that's about RCH. Now we'll be talking under section, like under this RCH, what are the different plans and programs or strategies we are following. Okay. So first thing is introduction of sex education in school level. So can you remember from which class you got the chapter human reproduction? You got the exposure of this topic of reproduction or adolescence in human being, right, from class 7th or class 8th. So Indian government has already incorporated this sex education into our elementary level. So students will be getting to know about reproductive organs. They will be quenching their curiosities. They'll be knowing about adolescence, why hormonal and physical changes or behavioral changes are happening during this period. And obviously students, as because they are the future of the nation, they have to know about the safe and hygienic sexual practices, how to avoid sexually transmitted diseases. Okay, so Kunal is asking, sir, Hind or update about Arjuna NEET English, or at least start date of batch. Already English batch launched, that was mentioned in app, and April 14, but English batch starting date mentioned April 13. So yeah, nearby that only the batch will be started. Okay, into the mid of the April. So don't like worry for that. Okay. Okay, for the time being, if you are enrolling into the Arjuna batch, you can check the lectures, quality lectures. They are from basic to advanced. You just have to check the playlist, and chapter wise you have to buy NCERT. If you are serious about your journey, about your preparation, you have to buy NCERT for biology, right? And you can check the lectures, right? Already you will be getting in the one shot section of PW NEET English channel. So that will be helping you, that will make you more advanced while you really will be appearing into the live classes. Okay, so let's move forward. Next motto of RCH was educating fertile couples. So why they're targeting fertile couples? Because they are the age group of the population belonging to, they can perform reproduction. They're at the peak time of their life cycle, they can perform this coitus and they can reproduce, right? So those married persons with people with marriageable age group, they need to know about birth control methods, they need to know about mother care, they need to know about postnatal care, means after the childbirth, how to take care of this baby and the mother, both physically and mentally and emotionally of the child and mother, and importance of breastfeeding. You know, in market there are n number of artificial milks are there, synthetic milks are there, and what is the difference between the synthetic milk and the real mother milk? You know that when a child is feeding on the mother milk, he or she gonna get actual IgA antibody of human being, actual human alpha lactalbumin, which will be helping the child to develop the immune system or boost the immunity. So that is the importance of initial breastfeeding rather than going for the artificial milks, right? Next, awareing or awareness about social evils. What do you mean by social evils? Sex abuse, rape kind of things, okay, discrimination in between sexes, right? Sex relative crimes, female feticide. Now, what do you mean by female feticide? It's a kind of old or orthodox kind of thought. In some villages in India, it was observed that if the wife is getting pregnant, then they will be that family will be sending them to doctor for the check that either the baby which is coming, upcoming baby is male or female, and they, if they used to find that the baby is going to be female, by the process of amniocentesis. Okay, there is a process called amniocentesis. By performing that, a doctor can like can guess can get the knowledge by studying the chromosomal setup of that fetus. If it's XX, then obviously the baby gonna be a girl child. If it's the XY, the baby gonna be a boy. So if there is to find that the baby is a girl, the fetus is of a female, then they have to perform forcible abortion, and that phenomena is called as female feticide. Okay, this is obviously a social evil, and government of India has already kept a statutory ban on this method. If a nurse or doctor is found to be performing this method, okay, then he will be subject to or sentenced to jail, as well as have to face legal actions, or if for the doctor, even the registration can be cancelled. Okay, so there is a statutory, means legal ban on the process of amniocentesis, because this is the process performing which you can get to know about the sex of the child. And under RCH, there is incorporation of massive child immunization program. You know about different vaccination like oral polio vaccine, measles and mumps and rubella vaccine, MMR vaccine, in huge level, in school level or in panchayat level or in municipality level, the vaccines are provided to the newborns, right? Okay. Now let's talk about amniocentesis. Now, what is the meaning of amniocentesis? Amniocentesis means surgical puncture of amnion with the help of syringe or needle, right? Why it's so? Why it's named so? Because if you check the fetus, or inside the, if you get inside the womb, this is the fetus or the baby floating inside a fluid called amniotic fluid, and that is covered by a sac or a membrane called as amnion. outside amnion you will be getting another covering that is called chorion. So outside the fetus or outside the embryos, human, we have four embryonic membrane, four extra embryonic membrane. The outermost one is called as chorion. Then after chorion we have amnion. Inside the amnion we have amniotic fluid, and inside the amniotic fluid the fetus is floating, and more two extra embryonic membranes are there. We call them allantois and yolk sac. Okay, but here we have to deal with this amniotic fluid with a syringe. A doctor or nurse will be drawing out the amniotic fluid, and you know that this amniotic fluid is containing cells of the baby. Amniotic fluid is containing the cells of the baby because the baby is floating inside it. Okay, now doctor will send this sample into the laboratory. There inside the laboratory they will be checking for the chromosomes present into the cell. So first you have to collect amniotic fluid, and inside these amniotic fluids you will be finding the cells of that fetus or cells of the baby. And if you check for the chromosomes or genes present inside, you can determine genetic disorder. You can determine genetic disorder. And that is why the process was invented, to determine, to search for genetic abnormality or chromosomal aberration in the growing fetus, like the disorders called hemophilia, sickle cell anemia, Down syndrome, right? These are examples of genetic disorder or chromosomal disorders. This process was meant for searching for this genetic disorder, but some unscrupulous doctors and medical practitioners, what they did, upon getting these cells and data about chromosome, they used to determine either the cells are having XX chromosome or XY chromosome. Then it is very much easy to determine the sex of the fetus, and this is called sex determination upon checking the amniotic fluid. And if they get to know that the sex of the baby is gonna be a female, then due to the pressure of the family, the wife or the lady is to be compelled to perform maternal termination of pregnancy or the forceful abortion. Okay, and that's the reason government has already kept a legal ban on this process, but with proper permission. If you are not going to check the sex or the chromosomal setup, you are going to check only for the genetic disorder, it can be permitted. Remember, the statutory ban means a legal ban against the detection of sex of the fetus. Okay. Another success under RCH, that is kind of a oral pill, that is an oral pill called Saheli. Okay. It's a kind of contraceptive pill, a pill to prevent pregnancy, right? Now, what's unique about this pill? It's a hormone free pill or non-steroidal pill. So most of the pills available in the market, they are actually either progesterone or progesterone derivative, or either combination of progesterone or estrogen. And you know that progesterone or estrogen or their derivatives are basically steroid hormones. Now if you are taking steroid pills as contraceptive, they are gonna harm you. They have very bad side effects, even cancer can be caused. That's why one institution called as CDRI, Central Drug Research Institute, situated in Lucknow of Uttar Pradesh. Okay, the scientists of CDRI, they had prepared a non-steroidal or hormone-free pill, and it's available in the market by the trade name called as Synchroman. And if a female is consuming that, there will be no side effect or very least side effect will be there. And generally what happens, those steroidal pills or hormonal pills you need to take continuously for 21 days. But if you are taking Saheli or Synchroman, you have to take only once in a week. Okay, again that's quite safer. And how it will be working, this Synchroman or the chemical component present into Saheli, they are acting as a estrogen receptor modulator. Now what do you mean by estrogen receptor modulator? Estrogen is necessary for thickening of uterine wall, and if there is thickening of uterine wall, then only there will be implantation of the embryo and gestation of the pregnancy will start. But what this Synchroman or the chemical component of the Saheli will do, that will modify the estrogen receptor of uterine endometrium. So estrogen cannot give signal to uterus, and if uterus is not getting chemical signal from estrogen, there will be no thickening of uterine wall, and if there is no thickening, there is no chance of implantation as well as pregnancy. So thing is that this Saheli can only prevent implantation, it will not prevent menstruation, it will not prevent ovulation, and that is why it has no side effect, because it is not directly interfering with neuroendocrine mechanism or endocrine system of your body. Okay. Now the topic is about population explosion and birth control. Okay. So there is increased health facilities and living condition. Okay. What was the motto of RCH? To take care of the mother and the baby, to give them a good life, as well as to control the population outburst in India. You know that at that time India was the second most populous country, but now in 2026 India is ranked 1 in terms of population, surpassing the Chinese population, right? So India is the first in the world in terms of or you can say the India is the most populous country. Okay, so why it happened? We already had a family planning program, RCH program. So it's happened due to this reason, due to increased health facilities. If you are getting a disease or discomfort, you will be visiting doctor, you have good doctor and good medical facilities, and you will survive, right? And we have good living condition, good health facilities. So that's why the explosive, the out rocketing of the human population in this world can be studied. So in 1900, the world population was near about 2 billion. In 2000, within 100 year, it has tripled from 2 billion to 6 billion, and within 11 years, 2000 to 211, the population of world was measured, that is 7.2 billion. Now let's take the instance of India. During the time of our independence, that is 1947, the population was 350 million, near about 35 crores. Now as of 2000, it has outbursted, increased into 1 billion. In 2011, the census, according to the report of census, the population was increased into 1.2 billion, near about 120 crore. And as of now, our population is crossing 150 crore. So that's called real population out rocketing or real population outburst. Okay. So what are the reason behind that? So a rapid decline in death rate, first thing. people are not dying. Average life expectancy has been increased. Decreased maternal mortality rate during the time of labor or childbirth. Mother is not dying, but in old days mother used to die sometime. And infant mortality rate has also been decreased, but there is increase in what? Increase in number of people in reproducible age are probable reasons for this. So you can say India is a country where majority of the population is youth. They are falling under marriageable age group. So more youth, more marriage, and more reproduction, and that's what making the country so much populous and reason behind the population outburst. Okay. Through our reproductive child health care program, though we could bring down the population growth rate, it was only marginal. Okay, so before the population growth rate was 2% per year. After implementation of RCH as of 2011, it's 1.7%, right? So that's why by implementing RCH, we were able to, it was able to decrease the population outburst, but it was only marginal. Only before it was 2% growth per year, now it's 1.7% growth per year. So according to 211 census report, the population growth rate was less than 2%, and that is 20 people per 1000 person per year. So that is the meaning of 2% growth rate. What do you mean by 2% growth rate? So suppose for a region, the total number of people are staying there, that is 100. So you have to take a span of 1 year. You have to take a span of 1 year, and after 1 year you will be observing that that particular geographical zone is having 102 people. So that is the real meaning of 2% growth in population. Okay. Now what NCERT is writing? If 2 people in 100, that means if the population is 1000, after 1 year there will be 1020 people. As simple as that. Now such an alarming growth rate could lead to an absolute scarcity of even the basic requirements. Okay, so nowadays you can feel the heat is because the scarcity for LPG cylinder is going on. Okay, the upcoming the current situation for our country. Why? It's because due to the outbursting of population. And even near future, if this alarming growth is sustained, then what will happen? There will be scarcity for food, scarcity for shelter, scarcity for clothing, in spite of significant progress made in those areas. Scarcity of jobs are already there, right? Okay. Next. So therefore, the government was forced to take up serious measures to check this population growth rate. Okay, so now what the government is doing, need to check that. The most important step to overcome this problem is to motivate smaller families by using various contraceptive method. And government is saying hum do hamare do, smaller families. Okay, even some literate population, some progressive couple, those who are staying in metro cities, they are adapting even one child norm. It's not even hum do hamare do, for them it's hum do hamare ek. Okay, so you might have seen the advertisement in the media as well as poster or bills showing a happy couple with 2 children with a slogan hum do hamare do, we too, our too. But many couples, mostly in the young urban working ones, have even adopted an one child norm. Okay. Now, population explosion and birth control. Okay. So, statutory rising, another method that is implemented by government. Legal rising, statutory rising, legally they have raised the marriageable age of the female, that is to 18 years, and that of for males, that is 21 years. If you are female and doing marriage before 18 years, and if you are a male, if you are doing marriage before 21 years, they can take you into the lockup, right? And inconvenience incentives given to couples with smaller families are two of the other measures taken to tackle this problem. So what are the measure? First, you promote the slogan hum do hamare do, even smaller family, one child realm. And second thing is increasing the marriageable age. So when NCERT was written, the marriageable age for male and female, it was 21 and 18. But for now, if you check it, the marriageable age for male and female for both, it's 21 year. Now let us describe some of the commonly used contraceptive methods which help prevent unwanted pregnancies. Okay, contraception. What do you mean by contraception? Contraception means the methods or the measures which can prevent a female to getting pregnancy, right? Prevention of pregnancy is simply called as contraception. Now what are the features? You have to look inside ideal contraceptive. Okay, so that contraceptive method should be user friendly, right? It should be easily available by your nearby medicine shop, and you can insert or use this by your own. You don't have to visit doctor or trained nurse. It should be effective. Like few contraceptive methods have high chance of failure, and the method you are using, that shouldn't have side effect. Like few contraceptive methods have side effect, a very dangerous side effect, like if you are consuming some steroidal pill that can cause cancer, cervical cancer, breast cancer, right? And the method should be reversible. So if someone is performing tubectomy or vasectomy, those are some contraceptive methods which are called regarded as irreversible methods. They are just cutting or operating their vas deferens as well as fallopian tubes through the mode of operation. So those are obviously gonna be quite irreversible method. But if you are utilizing condoms or diaphragms or some other physical barriers, if you are utilizing IUDs, they are reversible method. Okay, and if you are using those contraceptive method, if you are following the contraceptive method, it's not harming your sexual desire, it's not changing your sexual desire, it's not changing your behavior. So all these are the criteria. If you are getting contraceptive method, you are following a contraceptive method, if you are taking a device and that is having all these criteria, then it is said that that's an actually ideal contraceptive. Okay, so what are the different contraceptive methods can be followed by us? Okay, first we can explore about the natural or traditional method, then we can go with artificial method. Now remember that if you are going with natural or traditional methods, these methods are having high chance of failure, whereas if you are going with artificial methods, they are quite effective. But again, if we are talking about harm, right, side effects, right, upon using the contraceptive method, upon following the contraceptive method, this few of the artificial methods are very much dangerous. They have very bad side effects. But in case of natural or traditional method, they don't pose any harm or side effect for you. Okay, so there are three strategies we can follow: periodic abstinence, that is also called as calendar method, withdrawal method or coitus interruptus method, and lactational amenorrhea. Now, if you are thinking for artificial contraceptive method, we have barrier method. We have to use physical barriers, IUDs, intrauterine devices, oral contraceptive pills. The tablets or pills can be used and can be taken orally, injectable or implants, and surgical methods like vasectomy and tubectomy. Now, one by one, we'll be talking about them. First, it's periodic abstinence. So, what do you mean by periodic abstinence? Abstain or avoid coitus. Coitus means sexual intercourse. Avoid coitus during fertile window. You have to avoid coitus during fertile window, because what is fertile window? Fertile window is the period of menstrual cycle when there is highest chance of ovulation will be there. So, already I was discussing this technique. For a female, the menstrual month, the length of menstrual month is of 28 days. Means bleeding, when the bleeding starts, that is considered as the first day. Okay, and when the next bleeding start and the gap between them, that is of 28 days. So, we can consider the menstrual month for her is 28 days. Now, how to calculate the date of ovulation? So, you have to follow the ovulation calculator. So, you have to take the total duration or complete menstrual month, and you have to minus 14 with that. And if you minus 14 with from 28, you are going to get 14. So, it's the 14th day there is maximum chance of ovulation will be there. Is the 14th day there will be maximum chance of ovulation. Now, how to find the fertile window? To get the fertile window, you have to do plus 3 and minus 3 with this ovulation day. So, if you minus 3, you are getting 10th. If you are adding 3, then you are getting 17. So, 10th day to 17th day of menstrual month for this particular female is considered as fertile window, and if you are a couple, then you can track the calendar of your menstrual cycle or menstrual month, and within these 7 days, right, you have to avoid any sort of sexual intimacy or sexual intercourse. So, that's the meaning of periodic abstinence. Okay. Now, the second thing is withdrawal method or coitus interruptus method. So, it's very simple, but this method is having high chance of failure, and because it has to be maintained, or it has to be done by the male partner just prior to the ejaculation of the semen. Okay. So, just have to withdraw the penis out of vagina just prior, means just before to ejaculation. So, you cannot inseminate inside the vagina. You have to inseminate outside the female reproductive system, and that's called simply withdrawal method and coitus interruptus method. But remember, it is having the highest chance of failure. Another natural or traditional method, that is lactational amenorrhea. So, what do you mean by lactation? Lactation means milk production, and amenorrhea means absence of menstruation. Okay. And lactation means milk production and milk secretion. Now, how milk production is related to menstruation, right? Now, we'll be checking that. See, for milk production, you need a hormone called as prolactin. Prolactin. The another name of prolactin is LTH, a luteotropic hormone. Okay. Now, we know the common function of prolactin or luteotropic hormone, that will be influencing the mammary alveoli to produce, to synthesize milk. But another less known function but very effective here, that is acting as anti-gonadotropin hormone. So, prolactin can act as anti-gonadotropin hormone. That means high titer of prolactin can inhibit secretion of FSH and LH, because FSH and LH, these two are considered as gonadotropins. So, if there is the baby is feeding into a high frequency of the mother milk, the breastfeeding is going on. So, there will be high production of high titer of prolactin into the mother blood or mother body, and that will be giving a negative feedback or inhibiting the secretion of gonadotropins. If gonadotropins are inhibited, FSH and LH are inhibited, then what will happen? If there is no FSH, no LH, then there will be no ovulation. Then there will be no ovulation. If there is no ovulation, there will be no menstruation at all. And that's the meaning of amenorrhea, absence of menstruation. Now you got the point, right? Okay. So, here the main hero for this method is high titer of prolactin hormone in female blood. But one more thing you need to remember here. This is effective only up to 6 months after parturition. After childbirth, after the delivery of the baby, this is gonna work only after the 6th month. That is the limitation. Okay. Now let's jump into the artificial methods. In the first artificial method, that is barrier method. Barrier method can be called as physical barrier. Here we will be utilizing some physical barrier made up of rubber or latex. You can call them condoms, diaphragms, balls. Okay. Now, what this physical barrier is doing, the physical barrier will be prohibiting or inhibiting the sperm to get contacted or to get to perform syngamy with the secondary oocyte. So, on the way of sperm, you keep a barrier and sperm cannot touch the secondary oocyte. This is simple funda of barrier method. Okay. So, you are making a physical barrier in between sperm and ovum. Okay. Now, most common physical barrier, that is condom, and condom can be for male, condom can be for female. Okay. Let's check some government brands of male condom. It's Nirod. In normal shop you are not gonna get into, only you will be getting this Nirod brand into government hospital. A quite common brand for female condom, that is called as Femidome. Often female condoms are called as Femidome. and these condoms are made up of rubber or latex, and the advantage of using condom is that they can prevent sexually transmitted disease, because while a male or female is utilizing these condoms, it's covering the mucosa layers of the female genital tract and male genital tract, and there is mostly there is chance of transmission of the infectious agent which are causing STD, sexually transmitted disease, can transmit during the time of sexual intercourse through mucosa of male to through mucosa to female. As because these condoms are covering the mucosa layer of male reproductive tract and female reproductive tract, so there is no direct contact of fluid, right? And obviously one more thing, these are condoms are disposable, one time for one time use, okay. But this particular barriers called diaphragm, cervical caps and vaults, these are used only by females, not by male, okay. Now, what about this diaphragm, cervical caps and vaults? They are made up of only rubber, thick rubber is used, but for condoms you can use rubber as well as latex, okay. Condoms are disposable, but diaphragm vaults, these are reusable, and sometime to increase the efficiency of these barriers, some spermicidal creams or jellies or foams are used along with them, right. Spermicidal means the jelly or the cream which can kill the sperms directly. Now we'll talk about IUD. IUD means intrauterine device. Means why do you have to install them? You have to install these IUDs into uterus. PPT यही रहेगा, अभी 10 बजे तक तो जाएगा, 10:30 नहीं तो 10 बजे तक तो जाएगा, मैं घर जाता हूँ, उसको बताना होता है, बता दीजिए उसको. Okay, so full form or abbreviation of IUD is intrauterine device. You have to install these devices inside the uterine cavity, okay. And as per the NCERT, these are the most widely accepted method of contraception. When NCERT was written, IUDs were the most widely used contraceptive method, but nowadays it's these IUDs are generally getting replaced by condoms, you know, okay. So let's talk about different types or classification of IUDs. They are classified into three generations or three groups: non-medicated IUDs, copper releasing IUDs and hormone releasing IUDs. Okay, non-medicated IUDs are also called as first generation of IUD. An example is given written here, lips loop. And remember how this lips loop will be performing as a contraceptive. When you insert this lips loop inside the inside the uterine cavity, of a female, that will be stimulating the uterine endometrium, and it will be calling more phagocytic cell towards the surface, and that will increase the phagocytic activity of the uterus, and if there is more phagocytes, that can trap the sperms and kill. Okay. Now let's talk about copper releasing devices. Copper releasing IUDs, they are named as copper T, copper 7 and multilode 375. Now how they are performing their work? See here, means you can observe a copper T device here, and this copper T device will be releasing copper ions, into milligram format, right. Now these coppers are basically killing the sperm by slowing the sperm gradually. how these copper ions are acting as spermicidal agent. The sperms are in need of nutrients for swimming, and what is that nutrient? That is fructose. So what these copper ions will be doing, these copper ions will be blocking the channels through which a sperm can take, through which a sperm is taking fructose inside and supplying to the mitochondria. And that's how, when a sperm is unable to take fructose, unable to produce energy, it will become slowed down and gradually it will die, right? So that's how copper will affect the motility of sperm. Motility of sperm is affected. Motility means movement, capability of movement. And third generation IUD, that is hormone releasing, and you can see their name is given as per NCERT LNG20. Here LNG means levonorgestrel, a derivative of progesterone, and then progestasert. Okay, so this LNG20 or progestasert, both of them will be releasing synthetic progesterone. Either you can call those as progestogens or levonorgestrel. Okay. Now, if you want to insert copper T inside the uterus, so this T-shaped part is stuck with the uterine lateral walls, and this shaft of IUD is inserted, and you will be finding this fibrous thread kind of part. When you need to remove them, if you are like with the help of these fibers, you can pull them outside from the uterine cavity. Okay. Now, three different IUDs are exhibited here. So this particular IUD, this is called Lips loop. This is called as Lips loop, and how it will be working? It will be increasing the phagocytic activity of the uterine wall. This is the multi-load. This is multi-load 375. Now why it's named as 375? Because the surface area of this particular device is of 375 millimeter. That's why it's known as multi-load 375. Okay, copper T got the name due to the shape. Even copper 7 got the name due to the shape. But multi-load 375 copper releasing IUD got the name due to the surface area. Remember this. Okay. Now, this hormone releasing IUDs are releasing progesterone or synthetic progesterone, right? Now, what this progesterone is doing? See, progesterone is called is well known for its inhibiting activities of FSH, inhibitor of FSH, right? And if FSH is inhibited, ovulation will be inhibited. FSH is responsible for development of the ovarian follicles, and if there is no FSH or less FSH, ovarian follicles won't be developing and there will be no ovulation. If there is no ovulation, there will be no menstruation, and if there is no formation of uterine lining, ultimately FSH, absence of FSH will not produce estrogen, so there will be no formation of uterine lining, so there will be no implantation, and also high titer of progesterone in blood will change the quality of the cervical mucus. It will make the cervical mucus thick, so it will basically, what it will do, it will block the passage of sperms into the uterine cavity. So that's how progesterone works. Okay, now we'll be talking about pills or oral contraceptive pills. Now what do you mean by pills? Pills means tablet. Okay, and they are consumed orally. That's why you can call them oral pills. But there are few pills which can be administered through through vagina. So we will be calling those pills as vaginal pills. But in our NCERT, it has only spoken about oral pills or oral tablets. Okay. Now these are mainly, this can be hormonal pill, this can be non-hormonal or non-steroidal pill. We have already talked about non-steroidal or hormone-free pill, that is Saheli, right? Now we will be talking about hormonal pills which contain steroid hormones. Okay. Now hormonal pills can be of two types. First type, that is mini pills, and second type that is called combined pills. What is the difference between mini pill and combined pill? Mini pills are containing only progesterone or progestogens, means progesterone like component, and combined pills are containing progestogen as well as estrogen. So combined pills are mixture of progesterone and estrogen, mini pills are only progesterone pill. Okay, now how to use that? For 21 days you have to consume for every day, and just the 21st day is completed, you have to take a gap of 7 days. So during the time of these 21 days, while you were consuming, during the time of these 21 days, while you started consumption of oral pill, the progesterone titer will increase, and throughout the 21 days the progesterone titer will be maintained high into your body, and after completion of 21 days, for 7 days you won't be taking that pill, and that's the reason the progesterone will fall drastically, and fall of progesterone will give your body a signal, pill, please start bleeding or start menstruation. So after every 21 days you need to keep a gap of 7 days, and somewhere in between those 7 days there will be menstrual bleeding. Okay. Now when to start? When to start? What should be the first day? You should, if you are not taking, if you are a fresher, you want to consume the oral contraceptive pills, so when to start that? For that you need to follow this line: take within 5 days of menstruation. So if your menstruation generally is starting on the 15th of month, then what is that total range of menstruation? It should be 15th, 16th, 17th, 18th and 19th, first 5 days of your menstrual month. So from 15th to 19th, within these 5 days, anytime, any day, you can start taking this oral pill. Well, it's recommended, if this is the first day, if today your menstruation has started already, you can start taking from today only. That will give you a good result. Okay, some examples of these oral contraceptive pills: unwanted 21. Unwanted 21. Remember, there is another kind of pills are there called emergency pill. You might have heard about unwanted 72. So emergency pills are mainly combination of progesterone and estrogen, and after any rape case or unwanted coitus, you can take those emergency pills, and within 72 hours the effectivity of the emergency pills are quite good. Okay. Now we'll talk about another method of contraception, that is implantation and injectables. Okay. So these are kind of hormone containing strips is injected below the skin or below the gum with the help of special device, and they also contains the similar component like oral pills. They can contain only progesterone, they can contain progesterone plus estrogen combination. Okay. So they are similar to steroid pills, but these strips, these implant strips are containing progesterone hormone at a higher dose, and every day it's releasing progesterone or estrogen in a certain amount. So the longevity for utilization, the longevity for implants are higher than the oral pills. So if suppose for this month you have injected these implants inside your body, obviously it should be female, it's for female only. If a female is injecting these implants into her body, then the duration should that should be effective at least for 3 years or 5 years. Okay. Now uh, kind of irreversible method. Okay. Irreversible methods, you can also call them surgical method or sterilization. Now here by utilizing surgical technique, you have to produce cut into vas deferens in case of male, and the process is called as vasectomy. And for fallopian tube, okay, the pipe of fallopian tube, the initial at the region of isthmus, the fallopian tube is cut and tied in a perfect manner, and the process is called as tubectomy. So vasectomy is performed in males, the surgical cut produced into the vas deferens, and tubectomy is performed in fallopian tube, and that is for female. And why they are irreversible? Once you are performing this, there is very less chance that by again for the second time, if you are performing surgery to rejoin this vas deferens or fallopian tube, it will be very much difficult for the surgeons. That's why it said that it's irreversible method. Okay. Now if you are taking contraceptive, if you are using contraceptive pills or other methods of contraception, then you may face some bad effects. And what are those? Particularly if you are utilizing implants, if you are taking high dose of oral pills, then what will happen? You may face nausea, the feeling of vomiting, abdominal pain will be there, breakthrough bleeding. In between, anywhere in the menstrual month, there can be sudden bleeding into your uterus. So that's called breakthrough bleeding. Irregular menstrual bleeding, sometime for 1 day, sometime for 5 days, sometime for 10 days. So there is no proper length or no proper limitation. You can you are not understanding what's happening inside your body. Okay, it's not following the normal menstrual months. So that's called irregular menstrual bleeding. And chances are there, if you take too much amount of combined combined pills, is because they have high dose of estrogen, particularly these estrogens, high dose of estrogens can cause breast cancer. Okay. Now the next topic is maternal termination of pregnancy. Nearly 45 to 50 millions of MTPs are performed every year all over the world, which accounts 1/5 of the total number of total conceived pregnancies. Now this MTP is voluntary. You need to take consent from the mother and the family, and it's safe during first 3 months or first trimester. Okay, but still if you want to perform MTP on 4th month or 5th month, then you have to take consent, you have to take guidance from 2 medical practitioners, from 2 doctors, from 2 gynecologists, remember. So why MTP? Obviously the answer is to get rid of unwanted pregnancies. Remember, MTP is not a method of contraception. MTP is a legal method of abortion, right? So it's to get rid of unwanted pregnancies, either due to casual unprotected intercourse or failure of contraceptive device. So you were using a contraceptive device and performing coitus. But unfortunately, that contraceptive device was not effective. So in that case, you can go with MTP, right? Or for it can be prescribed for rape victims. MTPs are also essential in certain cases where continuation of the pregnancy can cause harmful or even fatal either for the mother or the fetus or for the both. If we are getting to know the fetus inside the mother womb is coming with abnormalities, maybe morphological abnormality, maybe genetic abnormalities, then under certain considerable grounds the MTP can be performed. Or even the pregnancy is giving emotional pain or harming physically to the mother is not taking in a positive manner. So under certain considerable ground, the MTP can be performed if mother is suffering from physical or mental or emotional agony. Next, we'll be talking about STIs, sexually transmitted infection, or STDs, sexually transmitted diseases, or venereal diseases, VD. STI is also called as VDs, or RTI, or reproductive tract infection. And we'll be talking about a few of these STI. First, gonorrhea. Gonorrhea. So it is transmitted or the infectious agent for gonorrhea is Neisseria gonorrhoeae, that is a bacteria. Next, syphilis. The infectious agent is Treponema pallidum, that is another bacteria. Genital herpes caused by herpes simplex virus, a virus obviously. Chlamydiasis caused by Chlamydia trichomatis. Genital warts caused by another virus called human papilloma virus. Trichomoniasis caused by Trichomonas vaginalis, another bacteria. Okay. Protozoa. Trichomonas vaginalis is a protozoa. Hepatitis B caused by a virus called HBV, hepatitis B virus, and AIDS, acquired immunodeficiency syndrome, caused by human immunodeficiency virus. Okay. So, out of these diseases, which are bacterial diseases? Gonorrhea, bacterial disease. Syphilis, bacterial disease. Then chlamydiasis, bacterial disease. Which are viral disease? Genital herpes, viral disease. Genital warts, viral disease. Hepatitis B, viral. AIDS, viral. And protozoan disease, that is trichomoniasis. Next, it is observed that the probability of getting infected is highest among these youths, or uh, those who have entered newly into the adolescence period, or in between the age group of 15 to 24 years. Now, HIV and hepatitis B, why they are special? They can be transmitted through sexual intercourse or coitus, but there are some other means, right? They can be transmitted like from mother to fetus or child through placenta. These viruses can cause can cross placental barrier, or during the time of breastfeeding, a mother can infect her child. It can be transmitted during the time of blood transfusion, or the infection can be transmitted through needles or surgical instrument. And three STIs are non-curable in nature. Which are they? Remember 3H: HIV/AIDS, hepatitis B, and genital herpes. Okay. Now, if someone is affected by STI, if you are diagnosing, so these are the early symptoms: itching, swelling, fluid discharge and pain into the genital region. And later complications, when it's severe, then there will be PID, pelvic inflammatory disease, inflammation and infection into the pubic region, pelvic region, abortion, spontaneous abortion chances there, stillbirth, a child uh, is already dead upon parturition, that is called stillbirth, ectopic pregnancy, pregnancy other than the uh, important or specific zones inside the uterine wall, then that is called ectopic pregnancy. In a wrong zone, in a wrong place of your uterus or in your body cavity, the embryo is implanted, that is called ectopic pregnancy. Chances of cancer and infertility. Yes, these are latter complications or severe conditions. This kind of signs can be observed by a doctor if you are suffering from STI. So, there is no reason to panic because prevention is possible except those three diseases, triple H: hepatitis, herpes and HIV AIDS. Okay. But if you are suffering from any other STI, one could be free of those infections by following the simple principles given below. First, avoid sex with unknown partners or multiple partners. That is the safest thing. Next, always try to use condoms during coitus, right? Next, in case of doubt, if you are having any doubt, one should go to the qualified doctor for early detection and get complete treatment if diagnosed with infection. Next, the next topic is infertility. Now, what is the definition of infertility? Infertility means inability to conceive if a couple is staying together for 2 years after 2 years of unprotected sexual cohabitation, then that condition is called as infertility. What are the different reasons of infertility? Maybe a physical problem, maybe some congenital genetic problem, maybe due to after a disease, maybe due to overdose of drug. If I talk about some disease, we can talk about anovulation in case of female, oligospermia, aspermia, azoospermia in case of male. The reason can be immunological autoimmunity is destroying the gametes, or sometime 70% of these patients are diagnosed, they really don't have any problem into their physiology, but they are suffering from psychological disorder. So, if psychology is treated by counseling, then they can get back their normal sexual life for 70% of the patients. Okay. Now, if you are really suffering from infertility, then you can take help of ART. What is the full form of ART? Assisted reproductive technologies. Here we need to take help of biochemistry and biotechnology. Okay. So, ART is of two types: in vitro fertilization and in vivo fertilization. Now, if fertilization is taking place into the laboratory, into the test tube, that is called in vitro fertilization. This falls under this program, test tube baby. And in vivo fertilization means when fertilization is taking place inside the female reproductive system. So, that is called as in vivo fertilization. Okay. Now, different types of in vitro fertilization we are going to talk about. First, we will talk about embryo transfer technique. they are ZIFT and IUT. What do you mean by ZIFT? ZIFT means zygote intrafallopian transfer. Okay, that means in artificial condition in laboratory, upon successful fertilization of sperm and ovum, zygote is formed, and upon zygotic development, when the embryo is less than 8 cell stage, then you need to install that uh, embryonic stage into the fallopian tube of the female, right, who want to be the mother, right. So that's called zygote intrafallopian transfer. So the point to be noted here, that is, you need to transfer 8 less than 8 cell stage of embryo. It can be zygote, it can be 2 cell stage, it can be 4 cell stage, right. Where it is injected, it's injected inside the fallopian tube. Now what is IUT? IUT means intrauterine transfer. Now, when the embryo crossed 8 cell stage, if you have a cultured embryo in laboratory which has crossed 8 cell stage, which is of 8 cell or 16 cell stage, which is kind of morula, so you can take that embryo and insert into the uterine cavity of the female who want to be mother. So that's called intrauterine transfer. Another technique is there, ICSI, full form is intracytoplasmic sperm injection. What happens here in the case of intracytoplasmic sperm injection? If this is the ovum, this is the nucleus, with the help of micro injection, will be uh, pushing sperm inside the cytoplasm of ovum. So that simple process is called as ICSI, intracytoplasmic sperm injection, and that sperm inside the inserted into the cytoplasm or ooplasm of the ovum will be carrying that another sperm nucleus, and they are gonna perform karyogamy and fertilization. Now let's talk about in vivo fertilization method. Here basically we are aiming for gamete transfer techniques, and when we go with sperm transfer, then they are called as AI, artificial insemination, and IUI, intrauterine insemination. For artificial insemination, sperm is injected into the vagina. In case of intrauterine insemination, sperm or semen is injected directly into the uterus, right. Now what about gift? Gift, full form of gift is gamete intrafallopian transfer. So gift is for ovum. Okay. So suppose a female is there, he cannot produce ovum because he is suffering from a disease called anovulation, but can provide good environment for the development of the embryo. She can be conceived. So in that case, the ovum is taken from another donor female and injected into her fallopian tube, right? And then in normal way, the fertilization and the gestation or the pregnancy can be caused. Okay. So that was all about art assisted reproductive technologies. So with this, I'm ending today's session. Good night, bye bye, take care and stay safe.